Hello, 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 hello.
Hal. नहीं नहीं, एक ही है। हाँ जी, आपको इस पे जाइए। हाँ ओके, अच्छा। हाँ इसी को करना है। हाँ तो अब की बार तो आ गया आपका। ये हमारा है। लॉग इन करूँ? अच्छा, पासवर्ड दिखाई कहाँ दे रहा है? अच्छा, हो जाएगा हो जाएगा। इधर नहीं लगेगा, उधर उधर लगेगा, देखिए। हाँ मुझे लगता नहीं है उधर, उसमें कुछ लगने की जगह ही नहीं छोड़ी है। तो हम इसमें देख लें। हाँ ठीक। मैंने कहा नहीं तो हम ये भी तैयार है कि हम इधर से देख लेते हैं, उधर प्रोजेक्ट प्रोजेक्ट कर दीजिए, वो भी चलेगा हमारे लिए। इसमें जो है आप अमरावती में पहुंच गए थे, 11 नंबर वाला। हो गया हो गया। तो थोड़ी बहुत एज यूजुअल, बहुत दिनों से नहीं आई थी प्रॉब्लम, तो अब आएगी। हाँ कॉमन वो है। हम्म, इधर। अच्छा, ये है ना, जो भी कुछ है। ये लीजिए, अंदर ही सारे पोर्ट्स रहते हैं ना, इनमें तो पता नहीं, नया है तो इनमें कुछ होता ही नहीं। हाँ ठीक है, हटा देते हैं इसको। तो इसको हटा देते हैं, ये तो इधर रख देते हैं कुर्सी पे। हाँ इधर रख देते हैं, कुर्सी पे रख देते हैं। यहाँ तो कोई बैठ नहीं रहा है, तीनों चीजें नहीं बैठ रही। सब कुछ उसी में रख दीजिए। हम्म, नहीं तो इससे इससे होएगा तो फिर ठीक है, फिर क्या जरूरत है ना। देखें, नहीं हिल नहीं रहा। अच्छा, अभी नहीं हिला तो खड़े होके। नीचे वाला ही अब आ गया, ठीक है, नहीं तो खड़े होकर हम कर सकते हैं, कोई दिक्कत नहीं। ओके, इसको भी रखें। Okay, so we will get started and yeah, mic on तो है, एक बार देखिए आ रहा है ना? Okay. So we'll get started, and today what I'm going to simply talk about is how the medicine for cancer has changed. Yesterday we talked about different therapies. This is how now people are thinking about how to treat cancer. Okay, and not all of it is immunology. There's a lot of nowadays a lot of new generation, next generation sequencing is involved in, or genomics is involved in the new treatments. And since, if you all are familiar, it has become much cheaper to sequence nowadays as compared to before with the new, this next generation sequencing. I think, I don't know how much it costs here, but the cost has been going down every year. So it takes about maybe $300 to $400 to sequence a sample, at least in the US. Here, it could be cheaper because we send the samples here, so or in China. So I'm hoping that it is cheaper.
They charge 50,000 something.
50,000 something. Okay. Approximate. Okay. Something like that. Okay. So, but in any case, the advantage is you can work with very small amount of cells and get a lot of data from it. And not always do you need very deep analysis. Sometimes for cancers and all, once certain set of genes have been identified, you need not do whole sequencing all the time also. So let's move on first. So in the old way of treatment, what you would do is you would try a combination of 2-3 drugs and see if the cancer is showing sign of shrinkage, or is it having some kind of an effect. And it was basically same treatment. It's a combination of 3 drugs you will give to anyone who comes. There was no difference between different patients. In the new model, now you base the therapy on the results of tests and genomics. So in fact, it is more of a predictable response. In this, you can predict that based on these tests and genomics that the patient will respond better, or should you use this drug or not, should you not use this drug. Okay, so let's see. Okay. So what you do is, these are all suppose patients, you do molecular profiling. Basically, maybe you look at the tissue, histology, what you do normally, biopsy. You also do genomics and maybe look at some other markers. And then you divide the patients, as you can see with the color. So you have prognostic markers, markers predictive of drug sensitivity resistance, markers predictive of adverse events. And then you divide the patients based on that. And then each one is given a separate therapy based on this kind of a combination of these markers. So why do you want to do this personalized medicine? Because it is more effective, and it has been shown that when you follow this kind of a treatment style, then there's prolonged survival. And ultimately, even though it looks as if you're doing more tests, you're doing genomics, it comes out to be less costly, simply because you're not using the very expensive drugs in many of the patients where that it won't work. So it has as I mentioned, the genomics and all costs comparatively much less than the chemotherapeutic regimen, because you have to give them, sometimes you have 4 cycles, sometimes 10 cycles, so you have to give a lot of drug. The other good part is the patient is not unnecessarily getting a drug which, because see, yesterday we had talked about chemotherapy. So all the drugs basically affect, usually are for cancer, of proliferation of cells, apoptosis of cells, or some of the signaling pathways which have changed. And none of these things are unique to cancer cells. All of these pathways, cell proliferation, cell death, are events which are occurring in normal cells also. So that is why you see so many of the side effects with chemotherapy. So if a patient is getting these chemotherapeutic agents which is going to do no benefit for them, it causes a lot of harm in the long run. So it has like multiple benefits is what I'm trying to say. So personalized medicine, people call it personalized medicine because now you are doing according to each person. So it does save lives. This is just to show you that 100 years ago, diseases of the blood like leukemia, lymphoma, and this is all these different ones. And today also you have all these different ones, but survival is changed from 0 to 70%. So it does make a difference is what I was simply trying to tell you. So this is another example where you have patients with colon cancer. So RAS pathway is a cell proliferation pathway which is very often mutated in many cancers. Okay, this is more of an example just to show you that you did KRAS testing and you found that these 4 people are not having that mutation in this pathway, while these have the mutation. So this is a drug which basically targets this pathway. So you will take these patients and treat them with this, while you will not treat these patients with this drug, because if they do not even have a mutation in that pathway, it is of no use giving them this drug. It may inhibit proliferation of normal cells and cause other problems. Okay, so you treat these patients with the drug, and now there is a higher ratio of treatment success also. And this is basically called informed decision making. So this is another example where you have a patient who is suspected of pancreatic cancer, and in the normal course what you would do is you would watch and wait, because you're not sure that this is really a cancer or not, or you will remove the pancreas. In both cases, it's not a very good choice, because if you are waiting and watching, pancreatic cancer is a highly aggressive cancer, so you have increased risk. If you have removed the pancreas, now you have insulin insufficiency, so you suffer from another problem, diabetes.
Ma'am,
Yeah.
why pancreas is very aggressive?
That I don't know. There are 2 cancers which I know, that pancreas and glioblastoma are supposed to be the most aggressive of the cancers. It could be that the mutations in them make them divide. Glioblastoma, it causes more problems because your brain, there's no place for the tumor to grow and go. So that I know, but I don't know why both of them are more aggressive. I just know that the survival time is much less for these people. So then we have, but if you do with the new approach, first you do this kind of a diagnostic. So usually these diagnostics are based on 2-3 different criteria. One is that you do maybe genomics. In the other, you also take the tumor biopsies and maybe treat them with different kind of drugs in vitro to see which of the drugs are showing an effect, and then treat the patient with that drug. And there are other methods also which we will come to. So if you find that it is pancreatic cancer, you treat aggressively. And if it is benign or it is not showing malignancy, then you don't do anything. So these are like informed choices. So this is basically just showing you that the survival rate based on these kind of modalities has increased so much for these diseases. Okay, this is just a data from different. So here is what I was talking about, that you have a patient. She has ovarian cancer, so you do a a genomics to find out if what kind of a mutation is there, and then identify the best drug for that mutation, and you do treatment. The other way also is side by side, you can also, so there is something known as xenograft models, which I will also come to, that you can put that patient's tumor in a mouse and try different treatments and see which causes tumor regression. Basically, you are using either an in vitro method or something to test out the drug before putting into the patient. That's the bottom line. And what model you use kind of depends on how much money you want to spend, because if you're going to put in mice, it's going to be more expensive than an in vitro testing. Okay. So why have we why are we now thinking of this personalized medicine? And this is simply because of the Human Genome Project, because with that now we know we have sequenced the whole of the human genome. So if you find out, now you can find out which genes are showing changes. Otherwise, you did not have that information earlier. You did not have this whole of human genome sequence. And the other advantage of genomics is, you hear about this omics word for everything. This is just to show you how many different omics are coming along every one day. Now metabolomics and everything is omics, right? So what I'm trying to say is that with genomics, the advantage also is, earlier you would know that, okay, this pathway, maybe this pathway is changed. So you are already biasing yourself towards those pathways. So you're going to look for only those pathways, because with the new next generation sequencing, when you align, you come up with many new genes which you have never thought of, which show changes, which you can study now to see how they are affecting. Because this is not something which you have thought of, okay, I know that KRAS will change, BCL2 will change, and this will change. So I looked for these three. I don't care about what is happening in the other genes. When you get a picture, whole big picture about what kind of genes have changed, what kind of pathways is it changing, it sometimes gives you very new targets, which may be more effective than older ones. So that is why people are very much interested in this next generation sequencing. So diagnostic technology has improved because of this. So past was macro level testing. Test differentiated disease from non-disease, and disease was defined by location and size. Okay, if it is in lung, we will do this. If it is in liver, we will do this. Now the division is coming more based on, okay, these type of all cancers with this KRAS kind of mutation, irrespective of where they are in the body, can be treated in this kind of modality. So it is no longer like, and today we do molecular level testing. So,
ma'am, when they do sequencing, they should have a control on normal cell and cancer cells, right? Then only they'll get to know what is the difference. If they just check tumor cells, even the same thing will be in the normal.
So usually you have a reference with the control.
Yes. From where they'll get normal cells. Suppose if ovarian cancer is there, it's basically small size.
So people who do not have ovarian cancer will be serving as normal, right?
Different control.
Yeah, and sometimes many of the times people make a database, like collect these and make a, like suppose how will I know that you are normal, right? So you take a database of at least 100, 200 people who are not showing signs of ovarian cancer, and then you do sequencing and you say that, okay, this seems to be the normal pattern, right? Because based on one person, how do I know you are normal, right? So that kind of a database people have established for many things because of all these things only. So disease is defined by individual biology or DNA of tumor or virus. Basically, it is easier to predict outcome, that okay, in this situation this drug will work, or not. So basically that's why this new molecular level testing is considered good. So this is just to show you that you can do multiple technology platforms. You can do multifunctional testing, multi-gene signatures as cancer. You can use different sample types like urine, saliva, breath. I've heard that nails also people have used for, because all cells have the same genomic DNA, right? So you can see all of us have, all cells in our body have the same gene signatures. Some genes are expressed in certain cells, some genes are not expressed in certain cells. That is there. But during sequencing, you will see that pattern in, so you can take from anywhere is what I'm trying to say. So will this work? Okay. Oh, sorry. I always, it's not showing the, if it is not, then we can move on, no problem. Click करें, छोड़ो, next पे, ठीक है, it's okay, we'll see it later. So, I was telling you that how you can put your patient's tumor in a mouse and then do a drug testing, right? So, these are known as patient-derived xenograft models. Because xenograft simply because the species are different, and you are taking tumor from the patients and putting them into, and these have to be immunocompromised mice, because in the normal mice you cannot do a transplant from human, right? So this is just to show you that that is what happens. You take a tumor expansion in PDX model, and then you can take that tumor and look for this high throughput drug screening. Or the other way is you try treatment A. So you have a cancer patient, you take PDX mice and you do one treatment A. You find that it doesn't affect the tumor at all. The tumor remains the same. In this case, you give a treatment B, the tumor is regressing. In this case, the treatment C, it is not doing anything. It's causing more toxicity maybe. So that's why now you decide that the treatment B is what you should give the patient. So this is again showing you the same thing a little more in detail, that you have a tumor from a patient. You can sequence also the tumor. So, and you look for what kind of changes have occurred. Here you find one amino acid change. Side by side, you are also putting the tumor in the mice and do a drug screening. So based on these two choices, now you have a database of genomic character, which drug outcome and biomarkers. So it will suggest which drug will work best.
Ma'am, we are putting tumor cells into xenograft, immunocompromised mice, right?
Immunocompromised.
So even if you put normal cell, it will grow because it is immunocompromised.
Yes, if normal cell can graft, I don't know but it won't grow to the extent as the tumor, because people do make humanized mice nowadays, where you take these immunocompromised mice and you put human PBMCs. Some they go at different places and they do survive for at least some time, and that is why now people are using humanized mice for some testing, because many a times what you find in mice may not work, right? And that at least they think it is human cells which are responding, and some of them do go to the spleen and the places where they should go. So that is definitely being done already. Like it's a lot of labs are using it. It's simply an expensive thing to do. So as I mentioned, that patient-derived xenograft models are in immunodeficient mice, right? So here is, just to show you that you have engraftment phase 1, phase 2, phase 3. Some people usually do research in this phases, and you can look for all these different things. metastasis drug screening. Now the new models believe that you should do orthotropic transplantation. And orthotropic transplantation means that if it is a lung tumor, you should implant it in the lung, so that you also, otherwise you were just implanting it in the skin. So now they want to see that if it is in the same organ as where it was in the original patient, so the microenvironment and everything is somewhat more similar. Do you see a better effect? That is, so basically, as we move along, we try to make things more and more advanced. So it is not, orthotropic models are more difficult because implanting in sub-Q in the skin is probably one of the easiest compared to putting it in the brain or putting it in the kidney or putting it in the lung. This is just to show you that this is a very simple thing, just to show these are immunocompromised mice and you're trying to put the tumor. So this whole process, if you see, again, even though it's showing so many nice things and you can do a lot of diagnostics tests with it, it is still a long process because you have to first get the tumor, you have to grow it in mice, then you have to test it. That will take some time. It is also an expensive process because you have to take these immunocompromised mice, you have to put the tumor, you have to grow, and you have to test. So and immunocompromised mice are expensive. These skid mice, because these are transgenic mice where you have done something to remove the T and B cells and other cells, right? So everyone, so these are definitely in use, but people are moving towards different newer things. How you can overcome this problem? How you can maybe make it more cheaper? And also the other thing is, you are putting a human tumor in a mouse, so there is that difference also. So this is just to show you. I think you need not worry about it. This is just an example of how different things have changed in how this model, the PDX model, is used in breast cancer, and what are the advantages and disadvantages. So they say that it is fast and cheap because that is, okay, I'll have to read it from here. I can't read this well. Okay. So if you do like a sub-Q thing, subcutaneously, then it is fast, cheap, and also it is technically more simple. That is what I mentioned. But the disadvantage is that it is an immunocompromised host, and not all. So it is not that anytime you put a tumor, it will grow. Some of the tumors just don't grow in the mice. So not all subtypes grow. And then tumor is growing at a peripheral site, right? And model is for advanced disease only, because wherever you can see the tumor and you can get a tumor biopsy, then only you can do this. You cannot do it in the very initial stages of the tumor where it's not even visible. So this one was the sub-Q. The next one is the orthotropic, and because breast cancer occurs in the mammary glands, so that's where they have put the tumor now that is orthotropic. So at least the microenvironment is appropriate. Tumor size can be measured because it's a outside one, so you can see measured with the capillars, and also you can even do surgery if you want of this tumor if it is orthotropic. But it is technically more complicated than the sub-Q model which I had mentioned. Again, the same problems are there, that it is an immunocompromised host still. So as I said, in all these, so metastatic one, in this, there's no primary tumor in the early stages, so that is one of the problems. PDX, in this case, only generally the aggressive tumors grow better, because I think they are more geared towards survival. So even in the immunocompromised host, they grow better, because ultimately you're doing a transplantation. It's not always going to work. So syngeneic is a mouse model, so like a mouse tumor in a mouse, so that definitely that should be fine. There's no reason. And then conventional, conditional GEM, these are, and the last one is what I had mentioned, the humanized models. These include human immune cells. Basically, in these mice, as I said, human cells are first, the mice are populated with human cells first, and then you implant the tumor. And but the whole process is not as easy as it sounds. So there is no standardization between laboratories, and there is a large variability, because each time you're in a mouse, you're putting some human cells from some individual. First of all, in humans in general, there's a lot of variability. So you're not using the same PBMCs all the time, right? So it takes longer, because first you have to create the humanized mice, and then you have to implant the tumor. And then it also relies access, you need the human tissue and human PBMCs, very high cost. Most importantly, it is very, very costly. So this, we will watch the videos maybe once we finish, then we'll just connect to the videos and watch. So to overcome this, you may have heard of organoids nowadays. Organoids are simply a 3D culture of your parts of your organs, because people seem to believe that when you grow cells on a monolayer, they are really not in their natural environment, and many of them adhere, right? You basically form a monolayer, right? So most of the time you adhere the cells to some kind of a tissue culture plate. So they, or even if they are in suspension, they are like single cell, which is normally not the case. So in that kind of way, situations, when you're doing drug testing, it may be different from what you are going to see in an organ or a 3D kind of an environment. So people now started working on organoids. So this is just to show you, this is a healthy organoid, this one, because the writing is low, so that's why I'm trying to tell you from here. This is a hepatocyte organoid. This is a tumor organoid, the blue one, and this is a apoptosized cell. So here you have a healthy organoid and you have a tumor organoid. You are doing drug screening, okay? And you find, and you are side by side also doing genomics, and you're then looking, you can use this for personalized medicine. So you now do testing of drug A on these. It kills both cells, the normal and the tumor. You see, both cells are dying. In this case, drug B, you again do the testing. Your normal cells are surviving and tumor cells are dying. That's what you want. When you do drug C, again the same thing is happening, that the normal cells are surviving and tumor cells are dying. So your drugs B and C are the drugs of choice. But now it seems what people have realized is many of the drugs show very more of liver toxicity, because liver is the organ which removes a lot of toxic chemicals, right? So side by side, they are now these two drugs which did not kill the normal cells, they run it on the hepatocyte organoid. So now they find that drug B does nothing to that, but drug C is killing it. So it means it is going to show, drug C is going to show liver toxicity. So with all this, what you have realized is that drug B is the choice you should, or drug B is the drug of choice which you should use to treat this particular patient. Because it specifically kills tumor organoids, it does not induce liver toxicity. At least this much of information you have gained from this kind of a culture.
How they make this organoid? They will put the collagen or
so there are many different ways. You will find nowadays even kits are coming which tell you, okay, take this, add all these different things. So some people do it in matrigel kind of a thing, where it is a gel like. So matrigel, a lot of people have even used for a long time, even to induce tumors in mice, because you take the human cells and encapsulate them in matrigel and then inject. Basically, matrigel has a property that when it comes to 37, it becomes more gel-like and liquefied, and so. But apart from matrigel, there is not one kind, because for each organoid there is a little bit of a difference. So it has a mixture of growth factors and some kind of a matrix is used. So I was mentioning matrigel, but nowadays new matrix are also coming. Some people use collagenase and fibronectin, and a lot of companies have come up with new kind of gel-like materials which seem to.
Because no, if you make a cell to adhere very tightly, then drugs penetration will be an issue.
No, in so the matrix are made in, these are matrix which are permeable, because you are also, they also, for the organoid to grow, it also needs the nutrients, right? So that's why the gel is such that it is permeable. It has, anything can diffuse through, and whatever you add goes through. It's not like it is a some kind of a solid thing in which you have embedded the organ.
Then this is better than a xenograft model, ma'am, in that case.
At least it seems so. In theory, definitely it seems so, but, and I think that's why it is catching on more. But see, with everything, whenever you do something, you will realize that even growing cells, I've seen that growing a cell line sometimes is very hard. So now you are talking about organoids. So you have to be highly trained, and you have, and these cultures are also longer. You have to do like 15, 20 days. So you need to be maintain sterility and all. You have to be very good at cell culture is what I would say. So, but still, I would say that it is a good model. So this is again an example. This is liver, and this, as you can see, is the colon, and you have taken a tissue biopsy dissection from that, and you have made organoids. So now you will see that a lot of, like if you look in the newer journals, you will see a lot of methods of organoid culture. So it is not, and people seem to be, but it is very expensive, because basically you need a lot of different kind of growth factors, mixtures of it, and nearly every day you have to add for them to be viable. Okay, so this, you are doing this organoid culture, and then you can make a digest, and like basically you are making a suspension of the cells, and then you can do clonal culture also, which can be, you can look for organ specific mutations. But I personally think that this testing is a better thing, because, and also you can look for intratumoral heterogeneity, because if you do single cell cloning, then how each clone is different you can look for by sequencing. You don't need to do single cell cloning. In this case, this is the element, the colon, basically the colon organoid, right? There's healthy, and this is tumor, both of them. And then you are growing them, you're making a single cell suspension, and you are testing your different drugs, which what we did in the last thing. And then in this case, what you're doing is you're looking at the mutation profile, simply because it's simply that whichever one is going to be more effective in this case, what is going to show more of an effect. And you also want to see if the drug you are using is going to cause a lot of mutation in the healthy tissue, because if that is the case, then you may develop new cells with more cancerous properties. Because if a compound is it causes a lot of mutations, then it can induce mutations in healthy cells and maybe make them cancerous. So this is a way of testing. This is different ways of testing, is simply this, what I will say. And as I said, we will talk about the, look at the videos later. So the third approach is now biomed engineering based approaches. So in this case, I'm just giving you an example here of something which is been done in our university. So in this, they have taken basically simply a small, this is a microfluidic based technology, and which they have just taken, like it's a device which is like a slide basically, okay, small slide. So channels are made in which they have seeded this fibrin gel. So that's why I was telling you that there are many kind of gels people are using nowadays. And this is fibrin, and now they have put, so I can read it here if you want. So in the gel, you already have cells. So that's why when you inject the gel into that channel, the gel also goes, the cells also go, okay? and defined location simply because they've made a location where you can inject. Otherwise, the channel is closed. You don't want the cells to flow out. Okay. And these are endothelial cells. So what happens over a period of time is it forms vasculature. Basically, it's forming these blood vessels. So it is like organoids, but in this case, you are starting from cells which are now, you're giving them the right signals, you're giving them an architecture so that it can form these blood vessels, basically. So, and here they have done this imaging to show that you can see these capillary formations which are occurring. Okay, and this is just to show you that how they have made a vasculature kind of a thing. Now this is the first step. Then the second step, see, you can read this paper and you can get more details, but the basic point what I'm trying to say is that you can do these small things in a small slide. And now what they did is introduced a tumor in this vasculature, because if you remember what I had told you yesterday was, which is shown here, this is the tumor basically, the green thing. What I told you yesterday is, what is a tumor? It's a mass and it needs to have blood vessels flowing through it to provide nutrition. So this is kind of a replicate of that in vitro. And many people have realized that you have to deliver drugs in the same kind of an environment. Now you have the vasculature there, you have the tumor there, and now if you do drug testing, it is more nearer to the way you would find in the patients, even though it is ex vivo. Okay, it's 3D.
Ma'am, if suppose if the tumor is the big tumor, which is highly vascularized, so what we can do, I feel, that we can load the drug into the platelet. Platelet is basically recruited to the inflammatory area.
Yeah.
So it will go and land there and then burst out. If it is bursting, it will not burst. So that we can deliver the drug.
So some people are trying platelet-based approaches. I don't know if for cancer, but for other this thing. But in cancers, many times a lot of platelet invasion is there. And some cancers, I think it is not considered a very good prognosis, because platelets have very high levels of this PDGF, platelet-derived growth factors, and they also seem to have a high levels of TGF beta in them.
So it is helping tumor cells to grow.
It helps tumors to grow. But when you engineer your platelets, at least you can make sure that they don't have these two things, right? And then maybe you can use them as drugs.
Here we are using it as a vehicle to kill the
cells. So people are using, because it's very easy to get a lot of platelets from very small amount of blood. In fact, it is, you take 1 ml and you can have thousands
anywhere,
of platelets.
All blood bank has.
Yeah. Lot of different approaches are being tried for delivery using cells. Some people take the red blood shell also, like leach out the hemoglobin. So now try to, insert something in the membrane of RBC. Some other people are using some of the cancer cell lines and using them as artificial antigen presenting cells. In fact, the reason I know about this research is I haven't done anything in this. Okay, I want to make sure, but I am collaborating with the person who has done this. And first, okay, maybe I can finish this and then I will come to that. So you see, they have put the tumor here. This is day 6, day 8, and then day 10. So it's growing. And then they did drug testing and they can see that it is changing. So this was like the very early, so in this they put a tumor cell line, okay, in the beginning, because it was like the initial thing and they wanted to test if their hypothesis will work. Now what they're trying to do in the same model is using it as that xenograft model. So instead of mouse, using this kind of a system, putting the patient tumor there and doing drug testing. And these are comparatively much cheaper devices, because these devices are usually made of PDMS or whoever is the engineer here will know more.
Organoid is better than this method, ma'am. Organoid, at least clumps of cells are there. So
in this also clumps of cells are there. Okay. So you can put a tumor biopsy. That's what they are doing now. So in they are introducing a
The
tumor
whole
biopsy.
tumor only, patient
Whole
tumor only
tumor,
they-
patient tumor, instead of making a mincemeat out of it. And side by side, here you have the advantage that the vasculature is also there. And you can deliver drugs via the vasculature. Because in general, if you are going to give drugs to the patient, it is
Intravenously.
also going to be via that. So, so it's not that this is better or that is better. I'm just showing you how different approaches are there and how you can, if you are an engineer, you would focus more on this. So a lot of work is being done on these small microfluidic devices. And when I'll do the immunoassay also, I will show you that now people are looking at cell-to-cell interactions using these microfluidic devices, because you can kind of make them in such a way that you have one cell here, one cell here. Now you can look at the different changes, and they can even take the cells from there and do single cell sequencing of those. So they can look at the direct effect of one to one, and a lot of different things are being tried with these microfluid device itself. Making the device itself is not that expensive. So, and you're not using that many cells also. So what I was mentioning was my collaboration is that what we were thinking is, yesterday I was mentioning that if you use things like dendritic cells or other cell-based therapies, the problem is these are personalized, like you have to take them out, load. So what we were trying to make were artificial antigen presenting cells, basically artificial, like 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 liposomes in a way, lipid vesicles having the MHC with the antigen, having the two co-stimulatory molecules, and giving it to, first we were going to try in vitro.
Pseudo APC.
Pseudo APCs. Will this work? Because the advantage is it's a very cheap method. You don't have to tailor according to a person. It is still very early, but a lot of people are trying different kind of other cell types sometimes. Some cell lines which don't express MHC, so they won't be rejected by the other person. They can be given universally. I think one of the example is K562. I think it doesn't express any MHC, and that's why it is used for NK cell killing experiments in the labs. So I think I was reading somewhere that they have expressed certain co-stimulatory and other molecules on the surface of these cells, and now they were trying to use them for as antigen presenting cells. So basically people are looking for approaches which do not require tailor-made therapy or this kind of, but if something is showing an effect, till we have an alternative, we have to go with that. It may take some time, I think. Everything, at least it's a start. So here is simply that with all these kinds of things, you have some advantages and some disadvantages. So embedded ex vivo tumor sections, this is basically like organoid cultures in some way. And what you find is that it maintains tumor heterogeneity. It is patient specific, but it does not have vasculature. That's what I was mentioning to you. Now you have 3D invasion models. In this case, sometimes this I haven't shown you, but it is just basically tumor is embedded in a gel, and then it is a 3D microenvironment. So as I told you, that people believe that when cells are adhering, they are behaving totally differently than when they are in a 3D kind of a system. Allows real-time tracking of cells because you can see them now. There are, in fact, the newer microscopes, you don't need to put any cover slips and all. You can put the whole flasks or plates, and you need not, they can show you organoids and all can be seen in 3D with these. And it need not be a confocal microscope which needs to take Z-stacks, or even if it does, but new ones are all automated. I think here also some people were looking at one yesterday, and there's also one from GIAN which does more or less the similar kind of thing. So again, this one also lacks vasculature and lacks tumor complexity. And here it is in 2D microfluidic device, typically for the study of migration. So this is a different kind of device. So microfluidic device people are also using for, if it is a 2D, if it's not like the gel-like structure, you put cells here, you put a chemoattractant here, you can see if it is moving or not moving. So that is also something a lot of people are using, and that is, I think even one of the companies is already selling it. Instead of the earlier kind of assay where you used to have a transwell, cells will be here, chemoattractant will be here, there's a membrane and through which cells have to pass. So those were assays of migration of cells which people would use. So in all these cases, if you see, most of the problem disadvantage which people believe is the lack of vasculature. So that's why when the other study was published, people were highly interested that it has the vasculature also. Basically, you have to understand this, that we are trying to mimic the conditions as much as possible. Every model has problems and every model has advantages. It's not that one is better than the other. Whichever works in your hand and whichever gives you answers is fine. So in the last 4 or 5 years, the exosomes, if you have heard of it, have also become very important. So what are exosomes? Here I am talking about exosomes and cancer, but exosomes in general have become important for many, many diseases. So the focus here is on cancer, but you can use this information for any disease of your interest. Okay, so this is just to show you that how many things the exosomes from cancer are involved in, and then we will go to what exosome is. So it is an activation of angiogenesis. It can do modulation of immune system. It can do remodeling of the extracellular matrix. It can do metastasis in different areas. It can re-educate the stromal cells. So it can do endocrine, paracrine induction of cancers. So basically, you can see that the exosome can be involved in all aspects of cancer. That's what I was just trying to show you with this slide, that it can have an effect on many, many different aspects of cancer, growth, metastasis, or anything connected with cancer. So what exosomes are basically, like your cells, they're constantly budding of small vesicle kind of things, and this was known for a long time. But more recently, what people have realized is that these small vesicles which are there, they contain exosomal cargoes. So they contain basically nucleic acids, like they have a lot of, especially microRNAs seem to be a lot more in these exosomes. They also have proteins, and they also have mRNA and DNA. And these are like a representative of what is found in the original cell, because they are budding off from the normal cell and they don't have a machinery to devise. But when they bud off, they take some of these material with them at the same time. And some of even presence of MHC1 and 2 have been shown found on these exosomes. So what people believe is that many times a cell is here, but the effect is somewhere else. Many of these effects are mediated by these exosomes, which are small vesicles, very small ones, 5 to 10 microns. They can travel anywhere in the bloodstream, and they can go anywhere, like even in the brain for that matter. So that's why so this is just to show you that this is the parent cell. These exosomes are budding off, which have all these different kind of things: RNA, pre-mRNA, DNA, miRNA, and then long non-coding RNA. In fact, every day there are new species of RNA, but miRNA have been shown for a long time. MicroRNAs can regulate functions of cells. They can either activate or inhibit, and these are now kind of a lot of inhibitory microRNAs and activatory microRNAs for different kind of things have already been identified. So, and long non-coding RNA is basically the non-coding sequences. Now people are thinking, earlier they were thought to be just be there for something, but now it is being realized that they also regulate gene synthesis. So they are becoming important, but I think they are very hard to study at this point. So these are proteins, and then these are ligands. So basically these exosomes which are coming off seem to have all these different things. and this is a recipient cell, so they can interact and release their exosomal contents. So if they have, suppose, an miRNA which inhibits immune, say dendritic cells. I don't remember the miRNA number, but miRNA 15 is there in large quantities. It goes and interacts. It will deliver the miRNA 15 in this cell. Then it can inhibit that cell. And even though this cell and this cell have never come in contact, but they are talking through this exosome and exerting the effect on the other cell, right? So that's why from cancer also many people believe that these kind of, all cells do this. It's a normal process. So cancer cells will also do this. So this is just to show that they can do metastatic things also, like they can take the cells and go for do metastasis also. But what I'm also trying to say is that a lot of people are thinking or trying to use exosomes for diagnostic purposes, because exosomes will travel via the blood, right? And if you can establish, and purifying of exosomes from the blood is already kind of established. So once you purify, you can maybe sequence and see before, maybe after treatment, the number of, the kind of exosomes in the blood change. Maybe that is so slowly you can develop predictive markers without going into the tumor itself. So this is what is known as liquid biopsy nowadays. Even for lung, now people, instead of going inside and taking a biopsy, maybe in the saliva or even in the fluid, you can purify the exosomes which are representative of your lung fluid. So you don't need to punch the lung. So this is why a lot of interest is there in this exosome. The other thing people think is, if these exosomes are going to go and interact with other cells, maybe we can load them. You can use them as a drug delivery system, because now you can load them with the miRNA of your choice, which goes and it works and it can inhibit something. So that's why there is, and as I said, and these will not generate any kind of an immune response, because these are normal. So it's like a very biodegradable system which can go anywhere and deliver, and you don't need to synthesize them as such. And as I said, purification is based on, nowadays, at least right now, because the field is still new, ultracentrifugation, because these are very, very small. So other things, if you centrifuge at very, very high speeds, these will settle down. So the purity of such kind of exosomes is about 80 to 90%, but you do lose a lot of them, because as with any centrifugation techniques, then people are trying ultrafiltration, because the size, that size will be from 50 to 100 microns. So you can devise that anything above 100 microns is cut off. And then
if you know the marker, we can sort.
So marker, they seem to have these tetraspanins on them. These are proteins like, if I go back, so these are 3-4 ones which are known to be present on this CD9, CD63, 81, 82. These are tetraspanins which people have identified over a period of time that they are present on most exosomes. It could be, it need not be all of them, it could be one or the other. And there are magnetic based techniques where people are trying to identify. The other way they are also trying to do is sorting. But it's simply that when you have large volumes, like exosomes are not always very large, in large concentrations in the blood. So you may have to process, say, 100 ml of blood. So then the other techniques become more difficult to do. Centrifugation or filtration is very cheap and easy to do. That's why. But both the other techniques, purification based on surface expression, it is already there. So basically, this is basically depicting what I had mentioned, that you can do different kind of things, basically deliver drugs, or look for, you can deliver different genes. Basically, you can use them as vesicles, right? So that is the advantage, but the other advantage is more for biopsy, for diagnosis, that it is being used. Because see, one of the problems with cancer is always you need to do a biopsy at the site. The other thing which people are trying to do is in the blood they try to look for circulating tumor cells, because they believe that the number of circulating tumor cells, if it is more, it means the tumor has metastasized more. That's why you're seeing, because the tumor ultimately has to travel through the blood vessels, right? So some of them will come, you will be represented in the blood. So, but the CTC thing, it is not the easiest of thing to do, because the proportion of these circulating tumor cells in the blood is very, very small. It is, I think, 10 tumor cells in 10 to 5 or something like that. So it's not an easy thing to do. Is what
And moreover, no, ma'am, anyway, it is already started traveling. There's no point in
it's
doing
simply,
research. And
it's more from a diagnostic point of view. And then now they're trying to see that how is it able to survive while traveling via the blood? Why is the immune system not attacking it? Because the tumor is not here in the blood, right? So tumor microenvironment is immunosuppressive, but the other parts of the body, why are they not able to kill these cells which are traveling? So now there is a new theory which people believe that these CTCs, they travel with the cells from the tumor microenvironment, like they form clusters with it and travel, so that they have their own kind of niche, which which is providing them, all the protection basically, in a way. So, but these are all like still hypothesis, is what I would say. So, so, so this is something which I read, so I thought it may be interesting to you all, is that now people believe that the gut microbiome, I told you that microbiome seems to be affecting everything, all the diseases. But then this is something which we know from long, right? That's why we have always been told to keep an eye on our diet. Diet has to be very controlled, and diet governs our elementary canal's health and ultimately the microbiome in that sense. But so what is being shown is that the gut microbiota, it seems to affect aspects of drug metabolism, basically uptake of the drug, anti-cancer effect, toxicity, and it even affects the absorption and bioavailability of many oral drugs, including cancer therapies. Okay, so sometimes it affects their mechanism of action, toxicity, and there are two best characterized drugs in this case, oxaliplatin and cyclophosphamide. And for both these drugs, the anti-cancer activity is affected by the gut microbiota, which primes the myeloid cells for production of reactive oxygen species in case of oxaliplatin, and facilitates the induction of anti-cancer cells in case of cyclophosphamide. So, whenever I have taken things from the journal, I think because it is sometimes it is not as clear, so I will read this. So this is local effects of gastrointestinal microbiota, and this is systemic effects. So here there is, bacteria produce a number of things. So there can be translocation of bacteria and bacterial products, metabolites. So the microbiome which is there in our gut, they produce a lot of metabolites. And I think I did mention this, but one of the examples is this short and medium chain fatty acids, and one of them which has been studied the most is butyric acid. And it has been shown that the butyric acid is produced when gut microbiome basically act on lipids. Butter and ghee is one of the things where it can act and make these butyric acid. And this butyric acid suppresses inflammation of the gut. So like butyric acid, it has been shown, even given in mice, in vitro also, it induces T regulatory cells and it suppresses inflammation in the gut. So people you can directly take. Yes, you can directly take, but then also you can take, it means you should not omit total, this concept of doing dieting and not having any kind of ghee or butter, everything is required. So omitting that is also not good, right? So if you have no butter, The bacteria cannot make
Yes,
these.
if somebody is
fatty
having
acids.
inflammation, high inflammation, they can take.
They can take. Otherwise, also moderately, if you are taking something, because your whole of the brain is basically made of lipids, right? So omitting that from the diet is not. I personally don't think it is a good idea. I think what I feel is that instead of omitting a particular class of food from a diet, it's better to take everything in moderation. But that is just my thinking. It's based on no science. Okay, so in case of in case of local effects, you have physiological functions. One is nutrient absorption. Oh, I forgot to mention. See, all the vitamins, bile acids, hormones, a lot of these things are synthesized by our microbiome. I'll give you an example of another study which I'm working with somebody. So they work with this B class of vitamins, you know, with the like this, biotin, thiamine, and all these. So, uh, thiamine, riboflavin. So basically, we have 4 or 5 of them. So specifically for biotin is what I'm going to talk about. So what they did is the biotin is not produced by us. Either it has to come from external sources, or microbiota also makes some of it. But there is a transporter in our gut cells, epithelial cells, which transports the biotin inside. Biotin is vitamin B7. So they made a mice which was deficient in this transporter. And what they realized after a month or so, these mice developed colitis-like symptoms. There was high inflammation in the cecum and gut. The only thing which was changed in these mice was that a particular biotin transporter was missing. And then when they gave back biotin, you can feed the mice biotin, the inflammation started to go down. So transport is not there, but it is not when you are giving via or injecting via the oral, if you're injecting or if you're giving via the oral route, then it doesn't need to be all absorbed by the alimentary canal. It will go also in the bloodstream. So even when, then they were confused because the transporter which is there transports both biotin and pantothenic acid. These are both two vitamins. So then they made diet which is only deficient in biotin and fed the mice. These mice are normal mice, not with the transporter deficient, and fed those mice this biotin deficient diet. Same thing happened, that they developed this colitis-like symptoms. So then they went back and looked into some patients also, and there seems to be some correlation between levels of biotin, and like colitis patients seem to have lower levels of biotin. So what I'm trying to say is the microbiome in our thing does so many things. So the other thing so it causes inflammation. My part was they wanted to see if this inflammation is coming from immune cells. So what we did is we just took dendritic cells, and we did a similar study on T cells. In both these cases, we took human ex vivo, like from the blood, and we cultured them in biotin deficient media, and then tried to stimulate them with, say, lipopolysaccharide or anything, if you say, but lipopolysaccharide in this case. They secreted much higher levels of the inflammatory cytokines. In the normal, where the biotin was regular, if it's here, this one was here. So it was just the deficiency of biotin was making the immune cells much more inflammatory. So this is another example that if the microbiome is producing this biotin, butyric acid, how important it is for maintenance of our, colon
Which
health.
bacteria is making the biotin, ma'am? Which bacteria exactly?
I think there are not one species, but many species of bacteria which make biotin, and I don't remember which one, to tell you the truth, because my job, as I said, was to look at it, and I didn't. Huh? Okay.
Ma'am, ma'am, we humans have more number of bacterial cells than our cells. Are bacterial cells controlling our immune system?
So what what is believed is, and it is known also, that they seem to provide a constant stimulation to our immune system, and also they are actively in the alimentary canal, they are actively inducing induction of Tregs, because again, as I mentioned, in the mucosa you don't want immune response against food, because that is one of the problems you have hear about these food allergies and things like that, because now you are starting to make response against the food, which is and in the lung also, with the air many things come, so you don't want to make an immune response, you just want to respond to pathogens which will cause disease, but not to things which are required for your body. So that's why in the whole of the mucosa there are ways where the immune system is kept in kind of check, and the microbiome plays a major role in that for sure, because if you change the composition, some of them are more inflammatory, some of them can cause disease, and those can activate the immune system. And I don't know the species he mentioned, he knows the species for the biotin one you were asking. So these are some physiological functions, and I think we have already discussed that it is involved in metabolism of bile and hormones, synthesis of vitamins, and fermentation of carbohydrates, morphogenesis, mucosal immunity I already mentioned. Then these are non-neoplastic pathology, so inflammatory so if there's it can lead to an inflammatory bowel disease, and ulcers are also, in that case also the microbiome is changed, and it can lead to stomach cancer. Heliobacteria you were mentioning is already known that it is associated with stomach cancer. And then you can now maybe play with this microbiome to improve cancer therapy, because now if you know that it can change the way the drugs will behave, maybe you need to play with the microbiome to enhance the action of the drugs. So here basically systemic effects are more or less same, some things are different, but we will go for the last one. So in cancer therapy, chemotherapy and immunotherapy and radiotherapy, efficacy can be changed by these microbiome. So this is an example. This is from that paper only I was able to find, which has come here, but it is basically that you have, it changes, if you take the drug, it changes the way the drug will metabolize. And I have it much better shown, I have a video which will show you that in a much better way than my trying to tell you with all these small things. So here you have these commensals. The video will come after this. This is Lactobacillus acidophilus, which I think is the most common one, which all of us know. I don't know if it's the most common one, but it is, all of us know. This is a gram-negative bacteria they are showing here. This is L. johnsoni. I need glasses for this. So basically these are other species of the commensal bacteria. So this is another class, and this is another class, and this is another class, is what they are trying to show. And this is, as you can see, is the intestinal epithelium. And this is the mechanism of anti-cancer therapy. So in this case, okay, this is untreated, the first one. In this case, you have patient DNA adduct, and then in this case, it is cisplatin or oxyplatin is given. I think I'll show you the video. I think this is going to make it more difficult for you to understand. So now we will first show this video, and then we'll go back to the other video. I have two of them, but we can do one by one. No, now it's connected. I did. Okay.
human body is made up of an estimated 30 trillion cells, but
Let it load.
it's also home to around 39 trillion bacteria. Together with fungi and viruses, they form a vast community called the microbiome. The largest and most diverse population of bacteria are found in the gut, with thousands of different types inside our intestines. These microbes may be tiny, but they play a huge role in our health. They help train the immune system, so it knows what to eliminate or ignore. They also help us digest food. But scientists now think gut bacteria play a role in how some cancers start, and maybe even how these cancers respond to treatment. Inflammation happens when the cells in our bodies become damaged or infected, and molecules released during long periods of gut inflammation can push normal cells towards becoming cancerous. Certain types of gut bacteria may be behind the bouts of inflammation responsible for starting some bowel cancers. And research has shown that the microbiome in bowel cancer patients can look different to the microbiome in people who are healthy. The latest immune-boosting cancer treatments have shown great promise, but they don't work for everyone. And scientists think the microbiome's link to the immune system could, in some cases, explain why. Research has shown that certain good bacteria are more common in the guts of some cancer patients who responded to immunotherapy. Scientists now want to see if changing a patient's microbiome might boost the effects of treatment. But exactly how these vibrant communities might influence cancer and the immune system remains a mystery. Right now, Cancer Research UK funded scientists are among those trying to find out more. And if they do, tinkering with the microbiome.
We can exit this one. I will show you the other one. That is a new study, and the researcher is talking about how they have shown with the microbiome a difference. And it's a longer than this, but it will tell you exactly. So that's why it's a so you can see how recent it is. It was just in August. So that's why I wanted to show you. And it is explained in a nice way in this
In this study, we focus in pancreatic cancer, one of the most aggressive cancer types, with a median 5-year survival of 9%. A minor subset of patients survive more than 5 years post-surgery. However, the factors that determine such enigmatic long-term survival are unknown. Previous studies have looked at genetic differences between those long-term survivors and the rest of the patients and found no differences. We have focused on 2 pancreatic cancer cohorts from 2 geographically different regions. We have compared long-term survivors, which are patients that survive more than 5 years, with an average survival of 10 years, with matched short-term survivors that live less than 5 years, with an average survival of 1.5 years. We focus on 2 factors, the first one being microbial composition of the tumors, and the second, the tumor immunomicroenvironment. Our findings were, first, we found that long-term survivors' tumors have higher microbial diversity, which means the composition of microbes in their tumors of long-term survivors was more heterogeneous. This suggests that tumor microbial diversity could be associated with benefit. Second, we found the tumor microbial signature that distinguishes long-term survivors from short-term survivors in both cohorts. This suggests that tumor microbial signature could be used as a biomarker predictive of long-term survivorship. Third, we found that long-term survivors' tumors have stronger immunoactivation, while the short-term survivors have more immune suppression. We then asked the question, can we modulate the tumor microbiome and immune suppression? To answer this question, we compared the microbial distribution in tumors and stools of patients with pancreatic cancer. And we also compare with adjacent normal regions. We found that around 25% of the composition of microbes in tumors is identical to the microbial composition of their gut. Therefore, this suggests that if we modulate the gut microbiome, we can also alter the tumor microbiome. We next collected fecal specimens from patients with pancreatic cancer, named in this slide short-term survivors, with long-term survivors and healthy controls. We transplanted these fecal specimens onto mice that had received antibiotics, and we then inoculated tumors. We found that the mice that received fecal specimens from short-term survivors or patients with pancreatic cancer had the larger tumors, compared with mice that received long-term survivors' stools or healthy control stools. Finally, we found that murine tumors' immunomicroenvironment were mimicking the respective fecal microbial transplant donors, with more immune suppression on the tumors from mice transplanted with short-term survivors' material, compared with more immunoactivation on the tumors from mice transplanted with long-term survivors' specimen. In conclusion, we have unraveled microbial events unique to pancreatic cancer long-term survivors. Due to the gut-to-tumor microbial crosstalk, fecal microbial transplant can modulate tumor growth and immune responses. And finally, we believe FMT emerges as a potential new therapy for pancreatic cancer.
So,
in this study, we focus in pancreatic cancer.
fecal transplant is also used in case of C. difficile infection. It is Clostridium difficile infection. It is known to cause it is a major problem in elderly subjects where it causes long-term diarrhea, and there is really no cure for it. And it has, I know that there are some studies already ongoing, and I think it is approved now that if you do fecal transplant from healthy subjects, you can cure these patients of this C. difficile problem. So I think I'm not sure, I don't even remember where I was, but let me just see. This was quite.
Ma'am.
Yeah.
Research are going to cure to cure cancer, but my question is that can we prevent our body from cancer?
So far I haven't seen prophylaxis for cancer. Like, usually people think that if you live a healthier life, you do exercise, you have less stress, you take a good diet, maybe it should be able to prevent most diseases. But so far I haven't seen any prophylactic vaccine or prophylactic like something which will prevent cancer. I haven't seen anything like that. I think one of the reasons is it is so heterogeneous. There's not a single reason why cancer occurs. So how to devise something which is going to take care of all these different modalities which cause cancer? That is a challenge in itself. And if we can do that, definitely I think it will be good for all of us. If we can prevent cancer from happening, then management is, so right now, but we are trying to manage it after it occurs, right? We have nothing which really tries to prevent it as such.
Ma'am, with this FMT, what are the chances of other pathogenic infection? Is there any?
So what I have seen is that this transplant, at least I know only for C. difficile, because that is something I attended a talk. This one is, as you see, it came in August this year. So I don't know. But what they do is they screen a number of individuals, and then they make a pool from those individuals where they do a lot of screening before they decide that, okay, these are the fecal transplants we will use. And it is from It was from like 50 to 100 subjects that they made a special healthy cohort which was screened for whatever different diseases we were talking, and those were the ones used for transplant in all patients. So that is taken care into, because that is definitely one of the things that maybe you're curing one disease but causing another one in that case. And also a lot of sequencing of these microbial diversity has been done. So now at least there are subclasses which now people realize seem to be more beneficial than the other. Even though more studies are required, but still.
But ma'am, every person have a different microbiome.
Yes.
Yeah? But there are also chances of immune response to that.
There are chances of, but these. So at least in C. difficile and all, what was shown was that this transplant need not be done like it was done like once every 6 months or something, and it was sufficient to get rid of that infection. It was not occurring again. So it maybe the bacteria, see, when you look at even in the whole of microbiome, many of them are highly immunosuppressive. Sorry, many of them are good commensals. So maybe if you're putting them, they don't. So commensals basically means that both your cells and these are living comfortably with each other. So if it is that class, then it doesn't cause an immune response. That is what I think. Okay, I think that was the last slide, and I have, if you want to see, then I have, I think, 2 videos also from before for xenograft and I think organoid, right? I'll just see. I have to go to the slide. We'll go 1 second. No. They can see this also, why not? One is, this is the personalized medicine one.
The fight against cancer is entering a new and exciting phase. Researchers and doctors are beginning to understand cancer and each individual cancer patient in far greater detail. These advances are enabling a much
Can you please turn off your Wi-Fi?
Oh, a lot of people are using it.
more personalized approach to cancer treatment. Cancer patients in the future will each experience a different, personalized treatment approach with the promise of significantly improved outcomes. What could the future of personalized cancer care look like? Meet Linda. Linda is a 62-year-old former smoker with a family history of lung cancer. Because her family history puts her at a higher risk of developing lung cancer, Linda's doctor sequences her DNA to test for genetic predisposition to this disease. This test reveals that Linda does have an increased risk of developing lung cancer. Linda sees an oncologist who prescribes annual checkups based on a technique called liquid biopsy. This non-invasive technique can detect traces of cancer in Linda's bloodstream and enable early detection of disease. Several years later, one of these liquid biopsy tests detects tumor cells in Linda's blood, indicating the possible presence of lung cancer. Unfortunately, further testing confirms that Linda has early-stage non-small cell lung cancer. Linda's oncologist orders several advanced diagnostic tests of her tumor, including multiplexing immunohistochemistry and next-generation sequencing. These tests reveal an abnormal pattern of protein expression in the area immediately surrounding the tumor, as well as the presence of several key genetic mutations. Linda's oncologist uses special software to help select a personalized treatment plan for her. The software compares Linda's profile, including her medical history and the results of the advanced diagnostic tests, to a comprehensive database of clinical literature and medical guidelines in order to identify the most appropriate treatment options. The software also compares Linda's profile to a massive library of data from other cancer patients, finding those people who had similar disease and generating insights from what happened to them. This sophisticated analysis identifies specific treatment options that Linda is most likely to benefit from, given her unique cancer profile, as well as a number of clinical trials in which she would be eligible to take part. Linda's oncologist carefully reviews these treatment options and their supporting evidence. She discusses these options with Linda, and together they agree on a personalized treatment plan. Linda begins a treatment regime of 2 targeted medicines that have been approved for use against her specific cancer type. Throughout her treatment, Linda's blood is tested regularly using liquid biopsy to detect response to treatment or disease progression. These tests show a positive response. Linda's tumor shrinks and after a few months is no longer detectable. Improvements in diagnostic techniques and treatment approaches, the collection and analysis of data from electronic medical health records, and sophisticated clinical decision support software will enable us to create increasingly personalized treatments for cancer patients like Linda. Personalized healthcare will help us to provide the best possible care for each patient. And that is progress in our fight against cancer. The fight against cancer is entering
Ma'am, how is the vaccine personalized for the treatment of cancer, for specific cancer treatments? Yes, yes, yeah.
लगा दिया हुआ है. Okay, we'll leave it. We'll not worry about it. So it is personalized because in that case you are taking out the patient own cells, then you are loading it with their own tumor, and then you are basically first differentiating them, loading with them with their own tumor antigens, and then putting them back in the same patient. You cannot do this, take cells from one patient and then put it into another. So it is personalized in that sense. But that one is more, I would say, a tailor-made therapy. There is no, it is not based on any kind of sequencing and thing. So vaccine, that is the only therapeutic vaccine. Even the term vaccine is used for that, it is not a prophylactic vaccine. So when you talk about vaccines, generally it is given before any kind of infection occurs to prevent the infection from occurring. But these are known as therapeutic vaccine, the cancer vaccines, because they are given after a disease has occurred and they don't provide protection, like it's not protection from, it's not a preventative.
Not like a, not precaution.
No, it's not a preventative medicine.
Is it like, it is not, it is temporary or it is permanent? The
Therapeutic vaccine?
yes.
It is temporary if because you have to do multiple rounds, and then it can be permanent if the tumor doesn't come back. Otherwise, you have to keep on doing this many
Multiple
times.
sessions.
Yes.
Okay. Thank you, ma'am.
Thank you. No problem.
Similar type of the organ from where it is extracted out.
Quite a bit, I think. At least a portion of it for sure. I've seen that people do this gut, they take a small portion of the gut, and at least in mice, I have seen that they grow it in small petri dishes also, in 6 well plates or even 24 well plates. I don't know why in this one I have maybe copied the wrong one, but I wanted to show you an organoid video, and that is what I was looking for. But every time I look at it, I think I copied the wrong link. Maybe I can find it online because I did a search and then
I'm asking this question because of the extent of any kind of the treatment. If we find out with the organoids, then where to go? This one is nothing but the in vitro type of the testing. Then where to go for the in vivo. But most of the time, what happened? Some specific type of the organoids, if we achieve the some kind of the treatment level, but it is not possible that it will extrapolate in the in vivo also.
not always, but there are 2-3 advantages. One is the organoid is going to be, so when you go in vivo, you are going into a mouse system, right? When an organoid is from a patient, so their own cells are there, right? So that is one advantage, that these are not mouse cells. In the other one, you're talking about the PDX model, which I mentioned, that you have to transplant the patient tumor into mice.
No, that was the extrapolation afterward. Before that one, the installation of any kind of the medicines that were invented one.
So at least it does tell you if the drug is going to be toxic for the tumor or not. From the organoid, if you saw, what it was telling you that if it is toxic to normal cells also, or is it toxic to only organoids. So at least you are eliminating, that will not change even if you go in vivo. The dose and other things may change. But if it is toxic, it will remain toxic, right? So that way the screening is faster, simply. And nowadays I think they do it in not even 6 well. I have seen that they have these 96 well. Yeah, and they have organs in them, these small, I don't know, very small organoids in them, and they do these testing. With high throughput only it is being done, I think. So
that means we can just go for the erasure of any kind of the wrong data which will
at
not
least
interpret.
you can eliminate, let's say you think that these are the 7 drugs which should be given to this patient. So you can eliminate, say, 5 of them. Now you know only 2 you can proceed with. So that is also a big thing in that sense. Like earlier you would have given all 7 to the patient, which would have resulted in different toxicities. In fact, nowadays many people are studying this, that if in patients who have been given chemotherapy, they have recovered and everything, but it seems to have an effect on the brain, which is which is visible for many years, and sometimes visible many years later. They call it the chemo brain, basically, is something, because these are drugs which are going to affect all over our body. So the minimum, it is always beneficial if we have to give less of them.
No, interconnected with this one, we are taking about the possibility of the toxicity causing to the organoids specialized with that organelle cells.
Yes.
But later on, when we deal with the in vivo studies, which are the other type of the tissues may also get toxic effect in that condition.
So in one of the slides I had shown you that they had taken this liver hepatocyte organoid, which was not a tumor cell site, and in that also they did the toxicity testing as a way to see that how is it affecting other tissues. Maybe we can also do that. We have organoids, maybe not from the same patient, because I don't think we can take biopsies all over from the same patient, but we can have normal organoids maybe from random samples, because, and hope that it will not, at least let's say if it is not toxic in a normal environment, there are chances that 70 to 80% chances that it won't be toxic in that person. Yeah, probability will reduce. Still, yeah, there's no way of saying that it won't be toxic because metabolism for each person does differ. So that is there. But I think, at least as I said, the probability of it being toxic goes down. I think I've either copied the wrong, because when I click on it, every time it's the same video which comes. So wait, I'm going to try the last time. Oh, mouse. Oh, ये वाला करते हैं. वही है ना? हाँ, ठीक है, छोड़ो. I'll show you. I'll send you the link for that. I think I have it in my other.
Ma'am,
Uh-huh.
from simple blood checkup, we know that can we know that the tumor is present in your body?
Only if you can find circulating tumor cells.
Ma'am, there is a checkup, 3-month checkup present, which know that which type of disease can occur in future. So can from this blood checkup, the different types of cancer like breast cancer.
So you're saying that you can predict using a blood test that 3 months later or 6 months later you're going to have that disease.
Yes.
I haven't seen any test like that so far, because something already has to change to be shown in the blood, and if that has changed, that means the disease is already started.
Ma'am,
yeah, WBC, but WBC increases in case of infections also, right? So how will you say based on that? That's why I didn't say. But not all WBCs will be beyond the normal limit, is it?
Ma'am,
in cancer, especially in cancer, the WBC increases above 1 lakh or.
But only in case of lymphomas and all that you see more of these. Yeah, but not in all tumors, at least. Not as far as I know. I'm not 100% sure, because I'm not like a physician and I haven't dealt in this area, so I should not comment on this. So, because otherwise, why do people have so much problem in detection? Only why is it detected at the late stage? In most cases, when the tumor is detected, it's either stage 3 and stage 4. You don't see the stage 1 and 2 half the time. Yeah, most of the time, people, when they develop some major symptom, then only they come to the doctor, and then you start thinking that it could be cancer. Yes, it can. So it has to have a certain mass, right, for ultrasound, for it to be visible. Yeah, so then it has already grown to that an extent that it is visible by the ultrasound. If you want to see something that early, then when it is 1 cell or 2 cell stage, then how do you see it? Is what because what he's saying is you want 3 months or 6 months before that, okay, you're going to get cancer. So that's what I don't understand how you will do. Yeah. Because
without pain, nobody will come to hospital.
Yeah, basically, most of the patients, there's something like a big growth, or if there is they have trouble breathing, if it is a lung cancer or there's some problem or the other, then only some people come. As long as the life is going on.
Is there any protein? Is there any protein which is available to detect, sense the size of the organ? So,
but the size of the organ is going to vary with different persons. So I don't know how you can make something which.
Because now, ovarian cancer, sometimes it will be like a football size. I don't know. There will be. Recently, I read on paper. It says in UK they removed 13 kg of ovary from one patient. So but what she was doing all these days till means.
See, this is simply the problem with us, right? We don't want
ignorance. Ignorance, no, ma'am.
Ignorance also, and then you don't want to accept half the time that okay, there's something wrong with you. You think that okay, some minor problem is there. If I'll go, then I'll have to take this, that. Why do that? It's it's just you don't want to do it. So maybe better awareness amongst people.
Mass screening, regular screening.
But so how expensive will it be? You think about it. Later screening only is so expensive. When you do mass screening, you're including all the healthy controls also in that. So
Ultrasound only will cost only 300, 400. If government gives 50%.
But ultrasound again, I said right, only when
size.
So ultrasound, and also how do you know where the tumor is? Because the ultrasound, okay, it is not whole body ultrasound, right? Human source. Yeah, but PET scan is also very expensive.
Yeah, but comparatively in India, they have exported, and the Panasonic is currently developing the PET scan, CT scan.
CT scan. Okay.
With the 3-layer mapping, including organelle, skeleton, and muscular structure.
So if there is any change, they can see. Because
near about the costing is for the one time the 70,000 to 75,000 rupees.
But then the timing has to be correct, right?
Yeah.
Maybe you had a PET scan to this year, and the tumor developed the next year.
Next year, it's not predictive that, but what he asking that how
screening.
To monitor.
Yeah, that
somebody is suffering from something, and he is not a healthy one feeling. Then he can try at least one
whole body scan, so that at least you see that.
Because ultrasound most of the time not able to interpret the everything in the organ.
No. Ultrasound, yes, it's not. CT will tell you at least if there's a rapidly dividing cell population. PET will tell you that, right? Because FDG, you can use like the glucose which I mentioned yesterday for mice, because that labels basically rapidly dividing cells.
Fluorescent.
And it shows up in the PET.
Bioluminescence.
Yeah. No, not bioluminescence. I think it is isotope 18 carb C or something like C18 or something like that. With FDG it is known as. I don't remember the full form, but it is used in PET scanning for that. And it basically lights up the area where glucose utilization is more. So it shows up. You can see the brain if you do the whole bodies. I have looked at mouse PET scan, so I can tell you this. You can see the brain, you can see the pancreas, because a lot of glucose is there. But in tumor cell areas also, because they also utilize a lot of energy, they are constantly dividing. So that they also light up those areas. So these two, you already know that it has to be there. So if you find it in other areas, then maybe you can suspect. But then also, still, the sensitivity of these techniques is very good, but still not to the level of 1 or 2 cells. You have to have some amount of mass which is there, like at least some, like even a small.
Suppose now if it is growing inside the intestinal lining, so the halo is there, space is there to grow. Patient won't feel anything. By the time it hits the other area, then patient will get constipation problem.
So then that's what I'm saying, so that till the patient gets any problem, nobody wants to go to the physician in general. At least that's what I've seen with myself and everyone else. So,
there is a anti-D globulin is given.
Yes, that is during the
pregnancy. First matter. My question is that why the body cannot regain or take the memory cells for the second maternity time. Most of the time that happen that at the first delivery, if there is a cross RH factor between the fetus and the maternal woman, at the first time they should reserve the immunoglobulin, whatever anti-D globulin is given. They should retain the that memories. But at the same, in a second maternity period, once again there is an administration of anti-D. Why?
Oh, so why you think that for the first time around, why is there not enough memory there being formed? So I don't know the answer, but I think the second time around, maybe the immunoglobulins are given more as a precautionary measure, that in case the memory is not there, and since you already know that this Rh factor problem will be there, so you give the immunoglobulin as a, if memory is there, fine, it may take care, but definitely if you give the immunoglobulin, it will take care, kind of a thing, precaution. Because if there is an immune response, there are chances that there's a very good chance that you will have the memory. And immune response is there against the Rh factor, and that is the problem basically. So either the memory is not enough, that's what people have realized, and that's why they think that you need to boost it along with the antibody therapy. So that's my take on it, but that's the logical part of it is what I think. Anything else? Okay, so maybe we will stop here, and then when we come back, I think many of you wanted immunoassays, so I will start with that, and then we go for other things. Okay.
Hello. 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 चालू आहे मोनो आउट फोन केलं. हॅलो. हॅलो. किती वेळ आला येत किती आहे. हॅलो. हा लेअर 5. हॅलो. हॅलो. हॅलो.
Hello. 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 Iraj maga ire.
So we will start the afternoon session, and this time I'm just going to talk about various immunoassays, basically the techniques you would use to study immune cells. And some of them may be already familiar to you. Flow cytometry I did in a special lecture, but these are some techniques are very common, like ELISA and which I think most of you are familiar. Others are how to look at proliferation, how to look at apoptosis and things like that, and then maybe some newer techniques also. So basically this first slide is telling you simply what, that different techniques which can be used in molecular biology and immunology. And, oh, क्या हुआ? ठीक. So we've been talking about that antibodies which are secreted by B cells, these are mostly polyclonal in the sense that even for an antigen, the antibodies are made against different parts of the antigen. So they recognize, so that's why there are multiple clones. Some may recognize this side, some may recognize this side, and some may recognize the bottom, basically any portion of the protein which is exposed. And that's why each B cell is different in that case, whichever one is. So it's known as a polyclonal response. It is not a single clone. So if you want to generate polyclonal antibodies, because you can use them for different purposes, what you do is you simply immunize animal or human subject with the antigen one or more time. One or more time simply because if you remember the immune response, if you do the first time, there is an antibody titer, but the second time around you will get a higher antibody titer because now the memory response is there, and also the antibody will be of higher affinity. You will get more of the higher affinity antibodies the second time around. So this is still used sometimes to look at, if a vaccine response is effective or not. But generally, this technique is, I would say, mostly used for that, or to look at an immune response for an antigen that people will bleed the mice or bleed the animal and look for if you are having the antibody against that antigen. So what are the good parts of this kind of a technique is that you can get different kinds of antibodies which bind different parts of the antigen. And these are good for agglutination and immunoprecipitation reactions. I'm not really going to talk about these two techniques much because these are mostly used in clinics and nobody really does anything in the lab with these two techniques. And the cons is simply because it is not a monoclonal, it's not one clone, you may find cross reactivity. And also the affinity of the antibody may vary depending on the time when you bleed. So if you bleed very early where the affinity maturation hasn't taken place, you may find the antibody affinity to be low, while later bleeds may give you higher affinity antibodies. Okay. I thought that it's okay. So I think in this slide, what we are talking about is that, I don't know why it is blank, but it is we are talking about that there is also antibodies which are monoclonal in nature. So monoclonal means only one clone of B cell against a particular epitope. Epitope is basically, if you have an antigen, whatever part the antibody is binding to is the epitope. It's known as an epitope. If it's binding here, it's the second epitope, third epitope. So it's just the area where the antibody binds. So if you want to look for only one clone of an antibody, the slide is missing, but Kohler and Milstein are two scientists who got the Nobel Prize for this. What they did is they took the bees, they basically immunized mice. Now mice will have polyclonal response, so it will have many B cells responding to different parts of the antigen. They took them, all of them, and fused them with a cancer cell. So what is the property of cancer cell? It's a myeloma which they fused with. The property of the cancer cell is that it does not die, right? So that's what they did. They because a B cell which is going to make an antibody will make for some time and then it is going to die. So then you are losing that antibody. So what they did is to make it immortal, they fused it with a cancer cell, and this cancer cell B cell fusion was still producing antibody, and they selected for the epitope they wanted, and now you have kind of a factory in a way. You can say this B cell is immortal, and you can produce loads of antibody because this B cell you can make it proliferate. People now do it even in bioreactors and all. So, and these are constantly secreting those antibodies. So basically you screen for the antibody you want and all those things, but it is like a antibody making factory. And this basically kind of revolutionized, you can say, the whole field of immunology in many ways. Because I've been telling you that, okay, this T cell has CD4 and you can use CD4 antibody to bind and recognize, but how do you get the antibody? Because each time if you were going to immunize a mice with CD4 and get CD4 antibodies which were polyclonal in nature, so sometimes they may bind this way, sometimes they may bind that way. Now if you have a clone which you know binds this way, and so it is better characterized clone, and you can produce it at any amount of quantities you want. So that is why this technique of monoclonal antibody generation was such an important discovery. And because of this only that now we have antibodies against nearly every possible receptor or intracellular molecule which we utilize for flow cytometry, or even when you look at microscopy or anything. In fact, for nearly most of the immune assays you will see you utilize antibodies. So monoclonal antibodies can be modified in many ways. One is that many people, what they do is, the antibody I told you is a Y-shaped molecule, right? And this region is constant. The top portion is where the binding specificity comes. So some people, because they don't want, so if you're using your antibody for, say, staining and all, the bottom portion which is constant, it binds to FC, receptors known as FC. So that can give non-specific binding. So some people just use the top portion, which is the Fab fragment of the antibody. So, and also, you know, I was talking about that the immune checkpoint and other inhibitor, other antibodies which are used for in cancer treatment and all. In that, they modify the constant portion and make it more humanized in that way. So at least only the binding portion is what has come from somewhere else, but this part is.
Ma'am, we are fusing the B cells and then this myeloma cells, myeloma. So my question is, why it has to unnecessarily express? Because if there is a antigen, it can produce B cells. How that cells are acquiring the character that it should produce antibody continuously?
Because when you fuse a B plasma cell with the myeloma cell, a B cell which is already specific for that antigen and is making an antibody, that's what you fuse with the myeloma. So basically, if you go through the whole process, if you immunize the mice, you will have B cells which are going to make antibodies against that antigen.
Yes.
So then you take that spleen from the mice which has those B cells, and it has antibody secreting plasma cells, and then you do the fusion. So they are already making the antibody, but now the only advantage is they are going to make it indefinitely, because now they have become immortalized. Cancer cells don't die.
Yeah, it won't die, but why it has to produce unnecessarily, ma'am?
What do you mean? Why unnecessarily? Because you are fusing it, and you the reason you're doing this whole process is because you want that antibody, right? Otherwise, why will you do this fusion? So that's why you want it. That's why you're doing this whole process of generating a monoclonal antibody, because you wanted it against, say, CD4 or CD8, whatever immunogen you immunized with.
So it will be under some oncogene, so that
no oncogene. It's basic. may involve oncogene, it may involve something else, but it's basically the property of why that myeloma cell has become immortal is what is going to be conferred on the B cell.
Yeah, I'm convinced with the immortality. Cancer cells has immortality, but why cell has to spend more? Why it has to produce this protein indefinitely?
So if it is not producing indefinitely, then each time you may get a different clone, right, which may not have the same affinity as before. So each time when you immunize, the B cell which is going to come for that antigen, it may not be the same as what came the last time, right? Because, so suppose I immunize this mice and then I immunize another mice. It's not that only the same clones in both are going to get activated. How do you know? Because 50 cells are, suppose B cells are getting activated in this mice, in that mice maybe 40 cells get activated. So, and in these 50, the one you want, is it there in the next mice, and how do you know it is that one only? And it has the same amount of affinity for the antigen. So each time then you have to go through the whole process of characterizing it, that, okay, does it bind properly? Does it bind non-specifically to something else? With the monoclonal antibody, once you have done the characterization, you can use it, because now you know everything about it. And each time you will use it, you will get a different result if you have a polyclonal kind of a thing. Here, okay, you know that it is going to give this kind of a result. So you are removing one heterogeneity, one variable from your experiment. Because suppose this time you are getting a signal, next time you are getting another antibody, you are not getting a signal. Now you don't know because the antibody is not binding, or the cell does not have the receptor for it. So that is why, and each time, how many times will you immunize the mice? Like how many mice will you immunize? Then you have to purify each time from the mice. So it doesn't solve any better, like that is not a better approach in any way, or even for that matter, a cheaper approach. Then how many mice are you going to kill? Because from one mice you may get for one experiment, but what about everyone else who is doing the experiment? With one B cell fusion, you are generating a monoclonal antibody which the whole world can use, right? So that is why the technique is so important. So you can change the monoclonal antibody in many ways. These are different ways which people have used. So because of the specificity of the antibodies, you can use them for a variety of purposes. And for whatever purpose you need to use, you can conjugate them with biotin, that is in case of ELISA. You can conjugate them with fluorescent dyes, which you can use for microscopy, or even FACS, which I was telling you about. Synthetic beads, you can use them for purifying cells. And I'll show you later, so I will not go too much in detail about these things. So this is, kind of before ELISA. First, radioimmunoassay used to be done. And in this, you would measure concentrations of biologically relevant proteins or hormones in the body. The basic method is more or less similar to ELISA, except that the radioactivity used to be used in this. And this method is slowly being phased out, because we are trying to replace most methods where radioactivity is used, because then you generate isotopes which live for very long. So I will move on from here, and something is wrong, I think, because it's it will take time. There's no video here. क्यों कैसे एफडी है? तो मेरा वो लेके आते हैं, इसमें कोई प्रॉब्लम है यहाँ पे? नहीं, तो इसमें तो नहीं थी आंटी, तो या तो सर से मेरा वो लेके आए, क्योंकि हाँ इसमें कुछ प्रॉब्लम हो गई है, या तो जो भी है, क्योंकि एम्प्टी स्लाइड्स तो मैंने बीच में इतनी सारी नहीं रखी है. Okay, but we'll move in any case. I think the next slide basically talks about ELISA. So ELISA is enzyme-linked immunoabsorbent assay. So if you want, say, if you want to look at some kind of an antibody, like suppose you are immunized with a vaccine or any kind, so then you should make antibodies against it. Now you want to see if that vaccine has resulted in antibodies being made. You can measure them using ELISA. So what you do is, in that case, you take a plate, you add whatever you have immunized yourself with. So let's say tetanus toxin in this case, because that is used as a vaccine, right? You can coat your plate with this tetanus toxoid. So basically, coating of a plate means the antigen binds the plate. Okay. Then you add a blocking agent. So because blocking agent is basically you add very high quantities of some protein or the other. Usually it can be BSA, it can be serum. Let's see. ऐसा तो नहीं है, मैंने देखा भी था मेरे कंप्यूटर में तो देख लेते हैं.
Sir.
We proceed and then we will look at that. So now how do I explain? Okay, so I was saying that you can, so if you're looking at an antibody in the blood, then you coat your plate with the antigen, and then basically you add a lot of non-specific protein, which can be BSA, bovine serum albumin, or human albumin, or some people even use milk, which has a lot of, and it's a much cheaper alternative. So basically high amounts of protein, which is going to bind anywhere it can bind. It kind of sit there in a way. It's not a covalent binding. And then you wash it off because this is going to sit there, and then you add your serum at maybe some dilution. So this serum should have a higher affinity for the antigen compared to what the non-specific, because that is not against that antigen. So that displaces the protein there and now goes and binds. So once the antibody has bound, again you do washing, and now you come up. So if it is from a sera from yourself, then the second antibody which you will use is going to be anti-human antibody, and that either can be conjugated directly to a enzyme, which if you add a substrate it will give color. So if you see this whole reaction, the antigen is there, the amount of antibody which is bound, the rest, the unbound, is going to be washed off, right? So only what is bound is going to be able to detect. So if there's more binding, you will get more color. It's directly proportional to what is going to be bound to the well. Let's see if it. ये देखो इसमें तो है, ये देखो इसमें है, हाँ, तो लेकिन ये पूरा नहीं लेके आए थे, तो इसमें तो लगेगा भी नहीं फिर, इसका तो वो चाहिए होगा नहीं, उसमें होगा, बैग में होगा, हाँ, उसमें जो उसका चेन है ना उसमें रहता है, हाँ, तो बैग ही ले आओ कोई बात नहीं. So there are many different. So either this, when the second antibody is there, you can have it already conjugated to the enzyme. And when the three enzymes which are most commonly used are horseradish peroxidase, yes, it is written here, and beta galactosidase, this is, as it says, rarely used, alkaline phosphatase, and horseradish peroxidase, HRP. This is the most common one which is used in ELISA. This is also used, but this is more commonly used. So the antibody, second antibody, is conjugated to the enzyme, and now you add a substrate, which in this case, if you use the okay, sorry, it gives kind of a blue color, and it is proportional because depending on how much the first antibody has bound, the second is going to be bound to that only, right? And you have washed away what is there. So if there is no binding, you will get kind of whatever the background color is there. That's known as the blank. But if there is binding, then you will get.
Ma'am, we are taking secondary antibody and conjugating with enzyme, no, ma'am?
Yes.
So how we are cross-linking that? What chemical is used to add the enzyme to the C?
Okay, I have never done that, and there are many methods which are available for linking these enzymes, but I'm not aware of what is used because I usually buy them. So, and this is, so nowadays those are so cheap, those antibodies, that it is not even worth thinking about trying to optimize that kind of a thing. It's too, it's a waste of your time, basically, is what I would say, because the second antibody is not even specific to any antigen. So it's any anti-human you will take, you can use it. So give me 1 minute. Make sure that I copy. इसमें करें, लेकिन फिर वो उसी नाम से आएगा ना, लेक्चर्स में कर देते हैं, इससे कोई दिक्कत नहीं होगा ना. प्रॉब्लम ही रहेगी. So for the enzymes, you have different kind of substrates. Some can be chromogenic, basically, which will give color. If you want them to be more sensitive, you can also use fluorogenic substrates. There. इसको प्रॉब्लम है, कंप्यूटर को प्रॉब्लम है, हम तुम बता रहे हैं, देखो फिर वही बोलने लगे, है ना, क्योंकि इसको कंप्यूटर में लेटेस्ट वाला खोल नहीं पा रहा है, क्योंकि 2007 है, वो 2016 पहुंच चुका है ना। हाँ। क्योंकि इसमें पूरा खुल रहा है ना, तो करप्टेड तो नहीं है। नहीं, क्योंकि फिर से वही चीज है ना कि ओके ओके विल मूव फॉरवर्ड। क्या है कि इसमें तो पूरा दिख रहा है, इसमें ओके मैं 97 2003 वाला करूं। कौन सा वाला? यही वाला ना। अच्छा अच्छा, कैसे पता लगेगा? ओके कोई बात नहीं, उसकी कोई प्रॉब्लम नहीं है, बाकी तो ठीक है। इसको नाम दूसरा दे दे, है ना, जिससे कि पता तो रहे कि क्या करा है। रीनेम कहाँ चला गया? कंपैटिबल कन्वर्ट कर दें। हाँ सेव एज तो कर ही दिया है, डेस्कटॉप पे किया है ना, तो और ये है, तो इसको नाम दूसरा दे देते हैं लोग।
2019 आज की डेट डाल के
25, चलो सेव हो गया ना। फिर से कॉपी कर लेते हैं, लाओ क्योंकि उसमें मुझे दिखे तो
ये वाला है क्या?
ये वाला है, देखा चांसेस इसकी ज़्यादा है, लेकिन कहाँ पर वही है ना। हाँ। तो नहीं तो इसी को लगा देंगे, एक की तरफ वीडियो चलाने की कोई खास बात तो है नहीं ऐसी, है ना, दिखेगा नहीं तो भी चल जाएगा, पढ़ के बता देंगे। कोई बात नहीं, वीडियो नहीं है इसमें। तो मैंने ऐसे ही कॉपी कर दिया है, तो इसमें कहीं पे भी मिल जाएगा। लेक्चर्स में जाना पड़ेगा मेरे ख्याल से, ऐसे
नहीं लेक्चर्स में डाल।
अच्छा से 25 नहीं नहीं, ऐसे ही करो, वापस जाओ, क्योंकि इसमें इस बार नहीं किया था, ऐसे ही से आई सर्च कर नहीं, ROI में चले गए हो ना, इसको मतलब पूरे उस पे जाओ वापस, जैसे कि हाँ, और पीछे जाओ। हाँ अब इसमें आ गया। हाँ नहीं, और पीछे जाओ, ये तो फिर एक फोल्डर में चलेगा। फोल्डर क्यों जा रहा है? उसमें वो कुछ कंपैटिबल नहीं है, हम लोगों ने बना दिया है, फिर अब ठीक से करेंगे, कोशिश करते हैं। देखो अब आ गया, ठीक। वो थोड़ा सा इधर उधर, हाँ, तो उसको हम लोगों ने ऐसा ज्यादा एडवांस कर दिया था, फिर पीछे करना। Okay, so we'll continue from where we started. So I mentioned monoclonal antibodies already. So this is just showing you the technique, that fusion of B cell with myeloma cell and then produces mass quantities of the single specific antibodies. And monoclonal antibodies can be used for a wide range of applications depending on the modifications we make. And we have already gone through this. So here is what I was trying to tell you. This is an indirect ELISA where you are taking, usually these are done in 96 well plates. So this represents kind of one well of that 96 well plate. And you don't use U bottom plate. 96 wells, you have to use a flat bottom, because when the machine is trying to read, it won't be able to read if it is not flat bottom. So you use you coat the antigens. Antigen coating is nothing. You make a solution of the antigen and you put it on the well, leave it at 37 for 3 hours or overnight at room temperature or 4 degrees depending on the antigen which it is there. So that, and these the plates which you use are special plates which have the high capacity to bind any kind of proteins, because most of the antigens you use are usually proteins. High capacity to bind things, I would say this, because then some of you will ask questions, if it is a carbohydrate, what will happen? But it can bind anything. So by overnight you allow it enough time for it to bind, and then you wash away whatever is not bound, whatever is excess, by using a simple PBS buffer. Usually the wash buffer which you use for ELISA contains very small amount of detergent. Usually the detergent is usually at 20, because you see in the next step, when I tell you that when you add this blocking antibody, the small amount of detergent which is there breaks off very weak interactions. So only a high affinity interactions remain. So that's why a small amount of detergent is added into the wash buffer. And this you can find in any textbook, any place, so don't worry about it. So then you add your specific antibody to be measured, and then you wash, and now you are adding the enzyme conjugated secondary antibody, and then you add some substrate which will give color here, and this can measure. So basically the amount of color is proportional to how much antibody is binding here. Because if 2 antibody molecules have bound here, this is just to say, then you will get this kind of a whole thing, then more color, because more enzyme is going to be there. Okay. Then the other method of ELISA is the sandwich ELISA. So this is very much used for detecting cytokines, because sandwich ELISA is used where in cytokines you have small quantities circulating in your blood, right? So you want to measure if, say, inflammasome is active or not, or even if you have activated your cell with something and you want to see if inflammasome is active or not in this cell. And if you remember, what I told you is that when inflammasome gets activated, it cleaves pro-IL-1 beta, and IL-1 beta will now be secreted. So if you want to, if IL-1 beta is there in the supernatant, that means inflammasome was active. So that is one way of measuring. So for measuring IL-1 beta, it is not an antibody. So then what do? What you do is you coat the plate with the antibody which is specific for IL-1 beta. Okay. Now you wash, and now in this case you add the whole supernatant, which if inflammasome was activated, it will contain IL-1 beta. or for that matter, you may have a control, you should have a control cell where you have not put that stimuli. That should not have an IL-1 beta. So if IL-1 beta is there, it will bind here, and then you add another antibody which is specific for this IL-1 beta. And usually in this case, you can have a direct conjugation of an enzyme here, but most of the times, and then you can get color. But most of the times, the amount of cytokines in the supernatant is very small. So to amplify the signal much further, most of the time, the second antibody, this antibody, has streptavidin on it, biotin on it. And then you add another layer, basically that is, the enzyme is not conjugated directly to the antibody, but conjugated to streptavidin, which is now going to bind to the biotin on the antibody. And the reason for doing that is, how will it amplify the signal? It can be 1:1:1, right? But one biotin molecule binds 4 streptavidin molecules. So that signal amplifies 4 times. So that is why you will see that most of the time the second antibody is a biotinated antibody, and then you come up with the streptavidin conjugated molecule, because that amplifies the signal. So even if you have small quantities, it is now become, the signal has become amplified 4 times. But otherwise, the basic process is still the same. You know, the washing, the blocking, and the color development, everything else is the same, just that you have amplified the signal. Then there is also a competitive ELISA. In this case, what you are doing is, you're taking an antibody with an antigen, and you have already allowed them to form a conjugate, like mix them together. So if they are specific for each other, they will bind to each other. And now and this is an antigen coated plate. Now if you add this antigen antibody mixture here, which is already complexed, if there is no free antibody, it should not be able to bind here, because there's nothing remaining. It's already bound to the antigen beforehand. And then you add enzyme conjugate. So in this, so basically the rest of the procedure is more or less the same. So this is usually used to measure antigen levels in pre-incubation solution. And I'll give you, okay, one thing I should show you. So this is, see, this is less color, more color, and this is most color. So this is blank, where say there was no antigen or anything like that. So usually you have a control, like in IL-1 beta, unstimulated cells should not give you any color. That's your control. So this is how the ELISA looks, and this can be measured by absorbance reader, which can measure the spectra at this wavelength. So depending on the color, the wavelength which you measure will be different. If in this case it is a yellow color, so you use one wavelength, but in alkaline phosphatase you get a different color, so you use that wavelength. So, but that is something you can just choose in the case of your ELISA plate reader has that option, depending on and so this, it's more or less what I have told you so far, that it can detect the presence and concentration of antibody in a sample. And this is usually, when you coat antigens, it is mostly to measure serum antibody. And this is an example that you can use this method to measure serum antibody against HIV proteins also. So these are usually useful for soluble cytokine measurements. And this also I already mentioned to you that sandwich ELISA is most common. In fact, most of the kits which you find, which are sold for the laboratory use, most of them use sandwich ELISA, unless you are looking for some serum titers or something like that. Especially for cytokines or any kind of protein mediators, because the concentration of these molecules is very small. So that's why you always need to. And this also basically says the same thing which I told you about competitive enzyme linked competitive ELISA. So when you are designing an ELISA assay, so unless you are buying a kit, if you want to make an in-house assay, then you have to, because that there are some proteins which maybe you are studying, which are not of interest for a wider audience, then you will not find a commercial ELISA kit. In that case, you have to develop your own. So the principle is still very simple, because it depends. What do you want to measure, the antibody or the antigen concentration? So the basic process is going to be the same. The only thing you may have to consider is how to get that antibody which is specific and works in ELISA, because if it is not sold commercially, maybe you have to raise it, or if somebody is selling it for Western, then you can use it also at that point. And this is the modification I was talking to you about, that you can, this is one way which allows kind of amplification, and also this people, you can use, you don't need to conjugate each and every secondary antibody with the enzyme with this, because if you just make biotinated antibody, you can use any kind of streptavidin conjugate. So if you want to do, the advantage in such a case is also this: if your secondary antibody is biotin conjugated, tomorrow you don't want to use an HRPO enzyme, you want to use an alkaline phosphatase enzyme, you can just take streptavidin conjugated to alkaline phosphatase. You want to do some kind of a fluorescent thing, you can take streptavidin conjugated fluorescent molecule. So it gives you an option to change the way you want to look at things. So that's why you will see that now most of the times it's very rare, and only in few cases there are available antibodies, but most of the times you will find that the secondary antibodies are biotin conjugated. And this is the flexibility which it provides, and money definitely, because it's, you have to do one kind of reaction, so it's cheaper. And this I think I already mentioned. So I told you that most of the time this is the method of choice or enzyme of choice, but alkaline phosphatase systems are used at a lower level and for safety concerns, because these could be carcinogenic, but now the new substrates have everything added to it. Earlier you had to add like small amount of peroxide to it, do this, that. Now all those things, there's a lot of changes have occurred, and the substrate which is provided is highly stable. So otherwise there was many problems associated with HRP or substrate. It will degrade over time, so it won't give color. But the newer batch of substrates which are now available everywhere are highly stable, quite stable. Not for 50 years, but they are stable for 2, 3, 4 years. So then when you're designing an ELISA assay, you should consider that your final readout, do you want it to be chromogenic? Chromogenic is where you get color using HRPO, right? And this is usually the most used because it is very comparatively cheap, because you need only an absorbance reader. So it's just like a spectrophotometer kind of a thing, which can only be in the visible region. You don't even need to have a UV region for that, right? So that's why it's a cheap option. The fluorogenic, it does give a higher, fluorescence is always more sensitive, but the problem also is it gets bleached, and you need to have a more expensive instrumentation to measure fluorescence. And also a lot of factors come into, then you have to change the plate which you are using, because if you are using a transparent plate, the fluorescent, the other well can get, if it's transparent, then the light can go into the other one also. So you have to use a black plate or a white plate. No, I think chemiluminescence you use the white plate. So this is a luminescence reaction where you, like luciferase, in a way you measure the light which is emitted in this. So it's mentioned here also, but in both these cases you require special equipment which is more expensive. In this also you require equipment, so it is not that you don't require an equipment, but this is much cheaper than these two. So nowadays with the new methods, so ELISA, from what I have told you, that if you want to measure one cytokine, you need to have a whole plate of it and you can measure only one cytokine. Now, with the advent of new things, now you can use that. So maybe I should go back. So you set up an experiment, and suppose you have very less amount of cells, and you get a total of 100 microliter of supernatant from that. You stimulate your cell, you have say 10 to 5 cells, some rare cell type, and you get 100 microliter of supernatant. You cannot do too much of enough. You can look at maybe half a cytokine one cytokine in ELISA, because you need to add 50 to 100 microliters for it to a well for detection, and you have to do at least in duplicate minimum. And if the concentration of that cytokine is low, then you may have to add the whole 100 microliter in one well. Okay, so there are new methods where you can use just 25 to 50 microliter of the supernatant, and you can measure 40, and now I think it has reached 60 kind of mediators which are secreted. It can be growth factors, hormones, cytokines, chemokines. All this can be measured in that 25 microliter or 50 microliter. Some kits use 25, some kits use 50. Especially for serum and other place, serum maybe not so much, because human serum you can get, to quite a bit of it, but still it's much more cumbersome. Suppose you want to do 30 cytokines, you have to set up 30 plates. You have to coat each one, you have to add for each one. In that 25 microliter of serum, you can detect all those things. So how has it become possible? This is simply because of newer technologies where now instead of the plates, they are using beads. Still the antibody is conjugated to the beads. It's not that the antibody is not involved. Same pair of antibody which was used for ELISA is now coated on the beads. So this technology was introduced by a company known as Luminex, so everyone still calls them Luminex beads. And these beads are usually now, I would say more in the recent years, they are mostly magnetic beads. Okay. Earlier they were polystyrene beads, and still the company sells those polystyrene beads, but I'll tell you why it is better in a minute. So this is a bead, and this has the antibody attached to it, and this is an analyte which has a different kind of cytokines. Now if you add this Luminex bead, it will bind. So you add a mixture of beads which are supposed to be against different cytokines. So if you're wanting to measure 40 things, you have in that bead mixture those 40 beads with all the antibodies. They go and bind to if those 40 things are present in your supernatant. Okay, so now, and then you again do the same reaction basically. You see, only thing is in the last step you have a fluorescent molecule, streptavidin P. Now, if you think about it, from what I'm saying, then you have taken 25 microliter of supernatant, you have added these beads with the antibody, and ultimately you have developed with streptavidin P, then how are you going to distinguish? Because it is like a one reaction. So it simply tells you that something is positive, but how do you know that what is the concentration of IL-10, IL-1 beta or TNF for each one? So for that, what they have done is that each of the bead has already PE, but at different concentrations, different levels of intensities. So based on that, they have made these bead regions. So the IL-1 beta will, if you look at the fluorescent intensity, the IL-1 beta has low level of PE conjugation already, and it falls in this 10 region, while TNF has very high level of bead has very high level of PE conjugation, and it falls in this 100 region, and something falls at that 40. So these beads are given numbers, and those numbers are associated with a particular cytokine, chemokine. So when you finish the whole thing and the machine reads it, then depending on the bead region and what is the intensity there, you can find out the concentration of each of the analyte. So if it is in bead region, say 48, and that corresponds to interferon gamma, and in that you got no color, that means in that supernatant there was no interferon gamma. while in the IL-10 region, you got, say, very high color. So you again have, so that means there was a lot of IL-10 in that. And I forgot to mention, both in ELISA and this, you always run a standard. Standard means like in ELISA, if you are measuring IL-1 beta, so you have IL-1 beta protein, it usually comes with it, and you know the concentration of that IL-1 beta, you make dilutions of it. So you start with 0 and maybe you are going to 1000 picogram. And you are doing the same reaction as you were doing. So now you know that 1 picogram gives you absorbance of, after your thing is finished, you find that 1 picogram gives you absorbance of 0.1, while 10 gives you absorbance of 0.1, then 1. So based on that, you make a standard curve. So basically the standard curve is usually like this, that at some point there is a saturation. So you usually go like this and then it is always saturated. So you take the slope region where it is showing linearity and you find out that, okay, 0.1 corresponds to 1 picogram from that graph. So in my sample, I have gotten an OD of 0.5. So it corresponds to 5 picogram based on this. So you can do this calculation. It's a very simple calculation. In this also, more or less, you also run a standard and you get this calculation is in the same way. The only difference is that you use these beads and you can use very small amount of sample to measure. And why the magnetic beads have become so important? So in like ELISA, you also have to wash these beads. So how do you? So it was becoming a problem. Earlier, they developed a filter plate where you add the beads, then you apply pressure from the bottom so that the beads don't go, but the liquid comes out, and then you wash it again. But with the magnetic bead, the advantage is they have this magnet at the bottom. So if you put the plate there, the beads stay. And yeah, and then with the magnet only, if you wash, it's easier. And the detection system, earlier the instrument, this Luminex instrument which was made, used lasers and it was very expensive. It was in the range of 100,000s. But with the new ones, what they have done is they have made these LED based detectors. So the machine itself is about even $10,000. So compared to 100,000, it is much, much less. But these kits which we use, depending on the number of analytes, they are expensive. So if you are trying to measure, say, 40 analytes, then they cost 3,000 to 4,000 easily. But the it's still cheaper than ELISA when you go for more number of analytes. When you go for 1 or 2 analytes, I would not advise that you do this. But if you are trying to measure more, then it is cheaper than ELISA, because for ELISA also you have to buy kit for each and every one of them and then do. So, but it is especially, now we are going miniaturized with everything. So like tumor biopsy, if you're getting very small amount and you're trying to grow, you cannot detect so many things which you want to detect. So in those cases, this Luminex technology is very, very important. In fact, some companies have started to develop gene detection based on this technology, but I'm still not very familiar with it and I'm not 100% convinced what they're doing. So that's why I'm not talking about it. So far we have been talking about ELISAs from the point of view of measuring in supernatant and things like that. But you can also use ELISA for measuring things inside the cell. And these kind of ELISAs are used very much for measuring signaling molecules as they become phosphorylated. So you can use a phospho-specific antibody to measure that. Or if the pathway has become activated and it has become phosphorylated, then you can use it to measure. So basically in this also you grow the cells in a 96 well kind of plate, and the method is more or less the same as what I told you for ELISA. And it's not that you can use in-cell ELISA to detect any kind of protein. as I said, mostly I have seen this to be used for signaling molecules, or transcription factors also I have seen that it is used. But mostly I would say signaling molecules, because see, if you look at the cell arrangement, so receptor is on the top, signaling molecules are mostly in the cytoplasm, and when you talk about transcription factors, they are in the nucleus. So to look at something in the nucleus, you need to permeabilize the cell quite hard, so that whatever antibody you are adding, it needs to reach the nucleus. Getting into the cytoplasm is relatively easier. That's why even for flow cytometry, when we do, we want to stain for transcription factors, your permeabilization technique is very, very harsh. Like one of the techniques which is used is a combination of methanol, 70%, and you leave the cells overnight in that at 4 degrees, and so that it dissolves away all lipids, and then the antibody will go in. But now, so that's just an example of what I was going to say. So in all cases, always, anytime you develop a method of your own, you always have to optimize it. That's a common thing. Even though it is written here for in-cell ELISAs, it's not something which is only for ELISAs. Now, another technique which is related to ELISA is ELISA spot. Okay. So in this case, you are measuring. So there you are measuring changes in signaling molecules using the in-cell ELISA. Here you are measuring if a cell is secreting something, some cytokine. Usually this method does not work for cytokines which are secreted at very small quantities. Generally, the ELISA spot is used for B cells which are secreting high amounts of antibodies, or T cells which are making granzyme, cytotoxic T cell, when they are highly activated and they are making granzyme, or T cells which are making interferon gamma. These are usually the most common. So in ELISA spot, the protein should be made, the cell should be making that protein in high quantities. Then it works more efficiently. With smaller quantities, the spots sometimes are, you don't get enough spots, or it's equivalent to the background. Because, so now we will go back and talk. So suppose you want to see if you have how many of your B cells in the blood are secreting. You were immunized with the tetanus toxoid, and how many of the B cells in your blood are secreting this tetanus antibody? Okay. First case, when you look at the serum by other ELISA, it will tell you, okay, you are making antibodies, that you have an antibody titer against ELISA. It does not tell you that out of 100 B cells, is it 4 B cells which are specific for your thing, or is it 6 B cells, or is it only 1 B cell which is making enough antibody that you are seeing? So for enumeration of the number of cells which are becoming activated or which are producing, you use this technique. So again, in this case, again you add the capture antibody, and you use a suspension of cells which are added to the plate, and if the cells are secreting that antibody, it will go and bind, basically, because you have something which will, if it is an antigen, it will bind to it will bind the antibody. If it's the opposite, it will bind and more or less, as I said, everything is the same, except in this case, the last thing, that it does not give you color like which can be measured by absorbance. Instead, there is a kind of filter at the bottom of the plate. See here, I can show you. So these plates are different. There's a filter here, which has the antibody coated thing which you have put, and so the number of spots which it leaves is equivalent to 1 cell. Wherever the cell has bound and produced something and is giving color, it is proportional to that cell. So here you had more cells which were producing that particular tetanus antibody. I'm just giving, that is an example. And here you had less cells which were producing that tetanus antibody. So this this technique, in many times people want to see that, as I said, that how many cells have become activated, then people use this. That the number of interferon gamma producing cells, in some cases, in tumors, it is a good prognosis if it has increased. But 99% of times I've seen it is being used in case of viruses where people want to look at number of interferon gamma producing cells, or in number of B cells which are producing an antibody. So similar to ELISA is another technique which is Western blotting. So in this case, this is a little more involved, like more work, I would say. In this case, you run the proteins on a gel, which will so this gel basically separates the protein based on molecular weight, because the gel is a mesh, so it allows proteins to travel based on the charge and the size. So based on that you separate, and then you transfer those proteins on a membrane, on a nylon membrane or nitrocellulose membrane, and then you do more or less what you do for an ELISA, because the protein, instead of the plate, is bound to the membrane, and now you come again add your antibody of choice to that, whichever protein you want to detect, you add that, and then do the blocking, washing. Basically you do the blocking first and then add and wash, and ultimately in the end again you add an antibody which is conjugated to an enzyme, and then you can do exposure to substrate. Earlier people used to use this film exposure, that they will emit this light, and now that is no longer used, that you would put your blot on the film, and since it was emitting light, the blot is what you used to develop. That is no longer being used. People now use either fluorescently labeled antibodies, and there are machines which can measure that. Lycor is one example, and it is more sensitive also. So depending on the methods, in some cases you can CCD cameras are also, CCD based technologies are also there, which can detect these different substrate exposure, the developed blots. Basically in this case what you will see is a band wherever the antibody has bound, and that you must be seeing in a lot of any journal. That's, I would say, the most common of the techniques which is used. So you'll see a picture of two bands like this to show you that if it has changed or more. So if there's more of the protein, the intensity of the band is more, and you can even, there are programs which can quantify how much of that is there based on the intensity. And also in all these cases you always run, in the Western blot you always use another antibody against a housekeeping protein, mostly like a beta actin. Like in PCR, if you do, you always run a housekeeping gene like GAPDH, because that's supposed to be your normal. If that is varying, then maybe the protein synthesis in that cell is overall low. That is also possible, that all proteins in that cell is in that lysate are low. So there are many checks you have to take, that you load equal amounts of proteins. You have to have antibody normalize against the housekeeping protein. So far we were talking about detection apart from the in-cell ELISAs outside of the cell. Now we come to microscopy, and I think a lot of people are already familiar with it. So again, you can use antibodies. So you have a mixture of these cells, and here if you see, blue is insulin and CD8s are in brown. So it tells you that the insulin secreting cells are separate from the CD8 cells, but CD8 cells seem to be attacking the insulin producing cells. If the two colors are on top of each other, then maybe the same cells which are CD8 positive are secreting insulin, which is not possible, but that is what it will tell you. So now So this still is using an HRPO kind of enzyme, and that is why it is giving you these kind of colors. But now in lot of cases, people use fluorescent labeled antibodies. You will see red and green or pink and blue kind of thing. I don't have it here. Okay, that I can show you from the internet anytime. Because fluorescence is more, it's more sensitive. So that's why it is used. And you can see see in this, because it is color, if it is only 1 or 2 dots, it is harder to see. Right now you can see because it is a large amount of protein. But for smaller amount of proteins, and especially some which are localized onto the membrane and receptor, fluorescence is a better technique. To use a fluorescent microscope is a better technique. So for subcellular structures, it is mostly electron microscopy because you need too high a magnification, and which doesn't work with other things with the normal lab microscope. Mostly for that you use this electron microscopy. I'll leave it here. And so this is the flow cytometry which I mentioned, right? So it's raining, I think, right? So this is what I mentioned. The immunofluorescence microscopy uses antibodies which are conjugated with fluorescent dyes, as you can see. So the confocal microscopy, how is it different from fluorescence microscopy is, if you take a slide and you put it under a microscope, it will take a picture. So it will show you only the top of it, whatever is you are seeing or whatever the microscope is seeing. Confocal microscope basically takes stacks, Z stacks you can say. So it goes in depth. So if you have a section which is say 20 micron thick, it will take two microns, 10 sections, or 1 micron. So it will take like 10 images, 20 images. And then if you can put those together, those are known as Z stacks. You can combine them and then you can get a picture of from top to bottom. So in some cases, maybe you see some fluorescence here. You may see a lot more fluorescence at the 3rd level or the 4th level. So in that way, that's why the confocal microscopy is good, because it gives you the whole picture, basically. So immunofluorescence has changed a lot so people have made even, these are mice are green because the GFP gene has been put in with the actin promoter. So generally I don't see this. Usually people use, some cells or the other which are labeled with the GFP, and then they can look, follow them where they are going. Even in tumors, people have engineered tumors, mouse tumors especially, that they have introduced GFP in it, and then there's a whole body fluorescence imager kind of thing also. So you put the mouse inside, so you can follow where the fluorescence is going. It's, GFP, rejection of the tumor.
GFP.
No, the GFP gene is transfected inside in these mice already in the embryo stage, so that all cells will express it. GFP is not a foreign protein for them as such. Green fluorescent protein does not come from mice, but it is not rejected, because you can transfect any gene you want into a gene. Is really not what is rejected. It's the MHC which causes the mismatch, and that is what causes the rejection, because it's the MHC which does not match between individuals, and that is what causes the rejection. So the immune response is made against the MHC, and usually the protein will not cause a problem. And this is the multi-photon microscopy, or 2-photon, or now in 3-photon also I have heard. And this is what I showed you yesterday. You can do 3D imaging live in mice, like whole intact organs. But in these cases, again, you need cells to be labeled from before. Because if you are going to take out a lymph node and you want to image under the microscope, just take the whole lymph node out or take whole spleen out, and if you want to image under the microscope, then how are you going to label cells? So the cells have to be labeled before. So in these case, people have made mice. In this case, I think the green cells are the T cells, which the mice is engineered in such a way that all T cells in that mice are green, and all DCs are going to be red. So then when you put the lymph node, if you see red, that means there is a dendritic cell. So similarly, so many mice have been made, like in some mice, they've made these are usually in the promoter region. So they find that all T regulatory cells are red. So you can follow them where they are concentrating. So that way, these kind of mice are highly useful. But in this case, I think I already showed you in the T and B cells. This is used to study interaction of cells. That okay, so far we visualize that DCs interact with T cells, but how is this interaction taking place? This movie will not run because it doesn't even run on my computer. But in this, it is like you can see this dendritic cell is presenting an antigen, and the T cells are coming and sampling it. They come, they go. They come, they go. Unless they find something unless the T cell is specific, and then it binds to the DCs and gets activated. Some of it you can see here. So using these approaches, a lot of different things have been done. People have imaged placenta, people have imaged lung. It is becoming more and more advanced is what I see. So each time a new mice is developed, which is some kind of a fluorescent cell to study these interactions. So it's like live imaging in a way. But because the cells are fluorescent, you can see. Otherwise, how do you see that these cells are, how can you differentiate between two cells? Two round things are coming. So that's why these techniques are, but difficult techniques. So this intravital imaging allows observation of immune responses in vivo. So similar as what I mentioned, that whole organs can be imaged, right? And this is a technique which is very, very useful for purifying cells from a mixed population. So in this, it's very simple. If you see, you take a magnetic bead and you conjugate it to the antibody. And suppose you have conjugated it to anti-CD4 antibody and you want to purify CD4 cells. You take human blood, you add this antibody, magnetic bead conjugated antibody. It will bind to, you add in sufficient quantities that it binds to all CD4 containing, CD4 expressing cells. So, and then if you put the whole mixture in a magnet, only the beads, all the beads will go to the magnet. The rest of the things you can wash away. With the beads, your cells are also bound. So now you have purified the CD4 positive T cells. You can also do an opposite kind of thing. Suppose you don't want to do a positive, this is known as a positive selection. Suppose you don't want to remove you don't want to touch the CD4, but you want to use them because you don't want that maybe binding of the antibody to CD4 is changing the cell in some way. So you want to use an untouched cell. Then you take an antibody against each of the other cells which are in the blood or whatever you're using. And you have beads specific for all those. You can remove all the other cells and you can use the remaining solution that should contain your CD4. And this is very routinely and commonly used. In fact, many a times people do this, that if they have a big volume of something, say blood, I have 100 ml of blood and I want to look at dendritic cells. Those are only 1% in the blood. If I go for flow sorting, where I can label those cells and the flow cytometer will sort them out. My problem is it's going to take me 5 to 7 hours to do that because flow cytometer has to run through 100 million of cells. Also, it is much more costly. And you use a lot of antibody. In this case, what I do is I do a pre-purification using these magnetic beads. so that I enrich the cell population. It's maybe 50% pure or 60% pure. It still contains a lot of contaminants, but from 100 million cell, I have reached about 3 to 4 million cells. Then I go for flow sorting. It can be done in half an hour, 40 minutes. And I get my very pure population, which is 90% pure. So many times people do this for enriching of the population also. So now we move to cell cycle analysis or cell proliferation. So basically, yesterday, I had shown you with the flow cytometry that you can label the cells with dye and the cells will divide. It will become less and less and less and less. This is a method which has been in use for a very, very long time. Simply, when cells are dividing, DNA synthesis is going to take place. So it needs to add the bases. So thymidine is one of the bases in your DNA. In this case, this thymidine is radioactively labeled, tritiated thymidine. So as the cells divide, this gets incorporated into the DNA. And then you can lyse the cells and you can look at the amount of radioactivity. It is going to be directly proportional to the level of proliferation, because the base will not get incorporated if the cells are not dividing, and more base will get incorporated if there's more proliferation. So this is very common method, and it is a very robust method also, and quite sensitive, especially when you have large number of samples. It is relatively easier to do than flow. Suppose you have 96 samples, you have to label 96 tubes, and then you have to run them through flow. In this case, you do in 196 well plate, and nowadays the harvesters are such, to measure that you can take the whole plate and put it a filter, and just that 196 filter is enough for it will tell you in each well what is the count. So it's relatively, I still think it is a good method. Then the other method is this tetrazolium salts. So it also basically more or less has same principle, that you add in a compound called MTT, and it becomes reduced when the cells are metabolically active basically. And then it forms in the living cells this purple kind of crystals, which then you can add methanol or something and it dissolves. So if there are more number of cells, more crystals will be formed, and more number of cells are considered that there's more proliferation. So this method is also used, but in general, in my experience, this method is very much less sensitive than thymidine or even CFSC. It's more, it's good, but it is comparatively less sensitive. BRDU is another method, and in this also, instead of thymidine, this also goes and binds to newly synthesized DNA. BRDU is phosphorylated in cells and incorporated into newly synthesized DNA in place of this dye, deoxythymidine. It's like an analog of this. Okay. So this is not a radioactive based assay, and then you use anti-BRDU antibodies to quantify the DNA. But it is, so we have used that method. It works very well. It's much more sensitive than the MTT assay, but it is more work than the tritiated. It's more work than tritiated thymidine, but still it is non-radioactive, so I would say it's a good method. In most cases, in our case, we tend to use more of the flow cytometry based assay, because as I said, if you don't have 50 to 100 samples, that is still an easier method. So this is assays of cell death. The radiolabeled chromium release assay is the first one which was used to measure NK. This was mostly used for NK cell or CTL to measure the cytotoxic activity of NK cells or cytotoxic T cells. Now it has been modified to use with flow. So because most, as I said, in most cases, what we are trying to do is remove wherever we have radioactive waste assays to reduce the generation of radioactive waste. So in this, so if you want to measure cell death, you can look at annexin-5 because when cells start to undergo apoptosis, the membrane flips. So you have the cell membrane up to, so this flips, the lipids on the lower side go up, and phosphoserine, phosphatidylserine is only present in the lower, in the inner membrane. So when cells start to undergo apoptosis, it flips. And annexin-5 can bind only to phosphatidylserine. So if you have annexin-5, you can, and annexin-5 is usually labeled with FITC or one of the fluorescent molecules. Now if you add it to the cells which you think are undergoing apoptosis, you should see binding of this annexin-5. If you don't see binding, then there is no apoptosis. And this tunnel detects DNA fragments. In fact, many people with the annexin-5 also use a dye known as propadium iodide or 7-AAD. These two dyes are different, the propadium iodide and 7-AAD, but both of them can intercalate, go into the DNA, but they can not penetrate live cells. Okay, so most time people use annexin-5 and 7-AAD. So usually there is a graph, I don't have it here, but if you look at the two axes, you see some cells which are annexin-5 positive, some cells which are positive for both PI and annexin-5. That means these cells have died completely because the propadium iodide has entered. And there are some cells which are not positive for anything. So if you have like a box here and 4 quadrants, this quadrant is cells which have not died, which are totally living because they're not positive for annexin-5 and PI. This quadrant is where cells are positive for annexin-5, so they have started to die. And quadrant where you have cells which are positive for both annexin-5 and PI, that means these cells are already dead. So mostly people use, along with annexin-5, the other dye also. Rarely I see use of it alone, because how do you know all cells are not going to apoptosize at the same rate, right? So that is the reason people use a mixture of both. So tunnel is another method which detects DNA fragments, because when cells apoptosize, the DNA also fragments. In fact, if you run the DNA apoptotic cell DNA on a gel, you can see it's like a ladder which comes. It's known as the DNA ladder for that matter. So tunnel is an enzyme which can be detected, and this is just showing you. BRDU is also for detecting cell death. I'll leave this here. And then the other assays which are for cell death is caspase. You know, which I was telling you that caspase is activated by inflammasome. It leads to generation of IL-1 beta, but it is also a signal of cell death. Caspases are basically, if they're activated, they ultimately lead to death of the cell. And several commercial assays are available. So basically, in this is the assay which is most used. In this case, there are substrates you add, and when if caspase is active in that cell, that means if the cell is undergoing apoptosis, it cleaves that substrate, and that substrate becomes fluorogenic, or it can be measured. It emits fluorescent light, which can be measured. And otherwise, people usually use Western blots also to look at caspase-based methods. So now this is the last part, more for the bioengineering, biomed engineering people. So now people are also developing microfluidic technology. One of the fluidic technology is flow cytometer. but microfluidic is miniaturized, usually in the form of a slide at the most. These are also being used to address immunological questions at a single cell level. So some of this I think I've already mentioned. So people are looking at migration, antibody screening, studying T and B cell receptors repertoire. I'm going to talk about the next one first, so that you can, in fact I will show you so many, I want to show you one more thing first. Yes, I'll show you this first and then I'll go back. So here, this is a paper which was recently published in Nature. Here they're just looking at the T and B cell interaction basically. You see that they have made a microfluidic device in such a way that one B cell pairs with one T cell, as you can see. And then they can look at this interaction. And what they found was, okay, I think I have, my slide is missing in this, but we have also developed a similar kind of technology. In, I told you earlier that we are also collaborating with some of the biomed engineers. So in this case, they are studying this T and B cell, but what we were studying is, we take one tumor cell and we put dendritic cells. So 1:1 pairing, again the same way. And we also have a way to measure glycolysis, or what kind of a metabolic changes are occurring in the tumor cell or the dendritic cell. And that you can measure by, there is something else which is developed by one of the other engineers in our university. So in the glycolytic pathway of ATP production, there is an enzyme where NAD gets oxidized to NADH. And that, or it's the opposite way, I don't remember. But whatever happens this way or that way, the substrate which comes is inherently fluorogenic. It is fluorescent. And they have developed this method that it can measure that fluorescence. So what happens in that thing is, this fluidic device where we have paired these two, you pass it through that machine. So it shows you what is, if this cell is glycolytic, or basically it measures the level of this fluorescence of NADH. And based on that, you can calculate if these cells are more glycolytic or less glycolytic. So side by side, we did like dendritic cells alone, tumor cells alone, and when we have put the two together, what is the change that is occurring? So what we found was that in tumor cells, which is, it is known that they are usually glycolytic. Tumor usually uses glycolysis for energy production. So we found more glycolysis. But what was more interesting is, we thought in dendritic cells, we will find less glycolysis, because tumor cells will suppress the function of dendritic cells. So similar to tumor, one thing I should mention here, similar to tumor cells, dendritic cells also use glycolysis for energy, especially during activation, because they have to respond very fast. So at that point, they use glycolysis. So if you see high glycolysis in a dendritic cell or a macrophage, it means that they are activated. So usually high glycolysis, low OXPHOS in oxidative phosphorylation in dendritic cells indicates activated dendritic DCs. So we were thinking that what we will see is that at the baseline, maybe whatever glycolysis is there, in this case, we will see less glycolysis. Instead, what we found was more glycolysis. So both seem to be becoming activated. But this is the study which only this much I can tell you, because this is what the results we have so far. But we are now trying to see that, is it because it was a tumor cell line that this was happening, and if you put a real tumor cell, then what happens? Or do you have to increase the amount? This was just like pairing for 3 hours or 2 hours. Maybe it is not enough to induce tolerance in DCs. Do you have to increase the time? Do you do it overnight, 2 days? Or does it change over a period of time? So those are questions which we still have.
What
to
is the
answer.
purpose of this experiment?
So this was basically trying to develop a device, first to see if we can even do this, and now we will replace it with real tumors, real cells from that patient, and then we can find out if we can, what kind of a change is there, because we are not labeling the cells in any way. So if you just take out tumor cells and you can put one, there were two purposes. First is you can just take the whole tumor and do this kind of an imaging and see if they are glycolytic or OXPHOS without doing anything. The other is you want to see if this tumor is highly immunosuppressive or not, right? And what is it doing to the dendritic cells? Then
so when you introduce the tumor cells to the dendritic cells.
Yes. Okay. So these are devices which are right now. So right now, suddenly, a lot of these microfluidic devices have come, which people are trying to develop to study different immune functions. So they are still very much in the developmental stage. So here we were trying to see how we can study this interaction. Okay, we can pair these cells. This fluorescent pairing is fine, but without touching the cell, what kind of measurements we can do? So